Very good morning to one and all present here to attend the morning session of fourth day of PhD Contemplational Conference. Today is speaking Dr. Ankit K. Rochani, Jefferson College of Pharmacy, Thomas Jefferson University, Philadelphia. His topic of discussion is personalized nano drug delivery strategies using smart and green biomaterials. I request all the delegates and participants to enjoy the session. Now I would like to invite Mrs. Disha to give the brief introduction about Dr. Rochani. Thank you.
Thank you, sir. A warm good morning to all the delegates, participants, and viewers. Myself, Disha Datta, Associate Professor, Devsthali Vidyapeeth College of Pharmacy. Once again, welcome you all for the fourth day of National Conference on the significant topic Recent Advancement in Pharmaceutical Sciences, funded by Power Finance Corporation Limited, New Delhi. During the expedition of last 3 days, Honorable Dr. B. Suresh, President, Pharmacy Council of India, and then we met many outstanding experts and experienced informative sessions. Now, for today's session, we are fortunate to have highly prestigious scientist and researcher, and he is all the way from United States. We welcome Honorable Dr. Ankit Rochani. His topic for today's talk is personalized nano drug delivery strategies using smart and green biomaterials. He is presently working with Thomas Jefferson University, Philadelphia, Pennsylvania, USA. Dr. Rachani did his postdoctoral from Bio Nano Electronic Research Center, Toyo University, Japan. He had his PhD degree from the same Toyo University, Japan, and Masters of Pharmacy from MS Ramaiah College, Bangalore. He did his bachelor's in pharmacy from Pune University in 2007. He was awarded Senior Research Fellowship in March 2014, funded by DST, Department of Biochemistry, IISc, Bangalore, India, Senior Research Officer in 2012, Sandor Proteomics Private Limited, Bangalore branch, India, and Junior Research Fellowship funded by DBST and DST, Department of Biochemistry, IISc, Bangalore, India. He also awarded with many honors like Embryo Research Grant Award from Toyo University, Japan in 2016-17 for the project Surface-Coated Magnetic Nanoballs for Cancer Therapy. Also, he was awarded with Embryo 15-16 from Toyo University for the project Multiple Drug-Loaded Magnetic Anti-Cancer Nanoballs. He is also the recipient of Japanese government scholarship in 2014, aced in professional courses such as NMR spectroscopy, proteomics for basic science and applied research, laboratory animal management, and basics of intellectual property protection conducted by Indian Institute of Science, Bangalore. His area of interest in research is design and development of smart biopolymeric nanoformulations for delivery of next-generation anti-cancer drugs, computational chemical biology, and molecular pharmaceutics for the development of drugs targeting age-related diseases like cancer, design of smart transdermal formulations for delivery of nanoparticles for cancer therapy, etc. His research work has been published in many national and international journals, having high impact factor, and also presented his work in different national and international conferences and seminars. He has also chaired different scientific sessions in national and international seminar and conferences. He has his also book written chapters. So I heartily welcome Dr. Ankit Rochani for his exhaustive. Please welcome.
Thank you, Dr. Disha. Thank you, Dr. Abhishek and Dr. Ashok, for this opportunity and presenting my talk. So I'll start with my talk immediately to save some time. Uh, I hope this is all visible to you all. So, so my talk. So I am Ankit Rochani, and Dr. Disha pretty much like briefly told you about what I've been doing so far, and I'm affiliated with Jefferson College of Pharmacy, and I am also a regional editor and a guest editor for MDPI Publishing Company. Specifically, I'm an editor for vaccines and polymer journals, which are four impact journal, and it's indexed in PubMed, Scopus, Web of Science, and all the other major databases, publication databases. So I'll start with my talk on personalized nano drug delivery strategies using smart green biomaterials. So to just give you an idea, so I am located in Jefferson College of Pharmacy, which is a part of Thomas Jefferson University. It's located in Philadelphia. So right now it's like midnight for me. So it's fine, but talking science is always fun for me. So I hope like you'll like this talk and you'll enjoy the knowledge that I am going to share with you. So Jefferson College of Pharmacy, we do lot of projects associated with LCMS, designing nano drug delivery systems, doing hydrogel rheologies, and also supporting Jefferson Hospital with lot of clinical trials in translation of some of the drugs that they have been experimenting from lab to the clinic. To add upon this, our lab like it's often audited by the external auditing agencies, hence it is a GLP compliant laboratory. All the data that is generated in our institution is either meant for the hospital or for some companies or the CROs, and some of the data is already in the public domain, it's already published, and some of them will keep on coming as and when the time goes on, so I'll focus like my attention towards the projects that we do in our lab. So my work is mostly focused on two main aspects. The first one is the formulation development and designs, and the second one is about studying the pharmacokinetics of these formulations and drugs in complex biological matrix like patient samples, serum, plasma, urine, saliva, tissue samples, as well as studying it in the preclinical stages when they are still not being translated into the clinics. But I won't focus much into the LCMS and the pharmacokinetics and proteomics and metabolomics area, but I'll be focusing more on the formulation part. So the slide in front of you there's three main components. I have mentioned like natural biomaterial. Just wanted to clarify, PLGA is a synthetic biomaterial, but I started my career and my work with PLGA. That's why I've just mentioned it over there. So there are three main components to this. One is the lipid, second one is protein, and third is polysaccharide. The reason I have been focusing on this thing is because of something called as developing a science of personalized nano drug delivery, where the idea is to use body's own biomaterial for making smart drug delivery platforms like nanoparticles, hydrogels, or nanofibers for delivery of drug molecules in the safest way possible. Up till now, people have been designing different types of or synthesizing new biomolecules, new polymers with different characteristics, but I prefer to use something which is coming from the body's own system, like proteins, polysaccharides, and lipids. So I'll be focusing as a lipid in the section of lipids. I will focus on PLGA and cholesterol as a biomaterial for designing nanoparticles. Albumin was used by my lab for developing nanoparticles as well as hydrogel systems. Cellulose is it's an example in polysaccharide segment where we developed water soluble cellulose for developing nanofibers for regenerative medicine. So to start with, I would like to go back like my days in Indian Institute of Science, where I was introduced to the fundamentals of heat shock protein 90. Now, I just want all of you to just step back and think about from a biological perspective, like why would you like to design a nanoformulation for a drug? So, for a biologist who is working on cancer therapy, he would be focusing, or he or she would be focusing on the molecular drug targets that are used for anti-cancer therapy. And heat shock protein is one of those hot and famous drug targets for treatment of cancer. But back in IISc, my lab was working on repurposing the technology of heat shock protein 90 inhibitors from cancer therapy to veterinary infections in India. And these were some of the major veterinary infections. So, like, and it was kind of a really, it is one of the most famous discoveries of my lab back in India. But I would like to focus more on cancer in my talk. So, heat shock protein 90 in cancer is overexpressed to overcome nutritional stress, proteotoxic stress, hypoxia, and genetic instability. So, heat shock protein is like a molecular, it's a molecular chaperone, which helps in folding the unfolded client proteins. So, like in biochemistry, the protein has to be in its folded structure to perform various cellular functions. But because the cancer is more of the tumor cells or the cancer cells are under proteotoxic stress, most of these client proteins are damaged. So to repair these client proteins, HSP90, along with its co-chaperones, help them to refold it and help cancer cells to perform its crazy biological functions that we think about, like metastasis or even causing problems like cancer-related deaths. So moving further, like the figure which I'm shown over here, it has like a heat shock protein 90 in the center, and all the client proteins like nuclear factor, transcription factors, and all the other protein kinases. These are the client proteins which are folded with the help of HSP90 and its co-chaperones and are responsible for carrying out functions like cell division, cell survival, energy metabolism, and all the essential biological functions for growth and proliferation of cancer cells. But the problem is, being a cancer biologist or a medicinal chemist, it's being taught to us that like we don't want the cancer to grow. We want to kill the cancer cells. So it was suggested by the professor in National Cancer Institute, and when I discussed this in my review articles, so the mechanism by which you can inhibit the HSP90 pathway. So the figures that you show rotating is an open conformational state of HSP90. So this is how it exists in the normal conditions. But when it comes in contact with the unfolded client protein, in presence of ATP as an energy source and co-chaperones, it folds, which is shown on the second diagram, which is rotating. And because of the folding, it stabilizes and protects the client protein, and voila, the protein is ready for carrying out its biological function. But if I have a HSP90 inhibitor, which goes and binds to the ATP binding site of the protein, the protein remain in its open conformational state, and it will never close. It will be never be closing it. As a result, the activation of the proteasomal degradation pathway happens. The unfolded client protein gets degraded. As a result, the cancer cell, would lose its important proteins that are responsible for its function. This is how the HSP90 inhibitor works, and this is how National Cancer Institute in the United States proposed various drug molecules that can be used as HSP90 inhibitors for the clinical trials, and they were translated to clinical trials, and they are still being explored. So being from the formulation background, I got fascinated with this idea of the HSP90 inhibitors, and they were extremely. It was a boom that many companies were investing in discovering and developing various types of HSP90 inhibitors with different structures. So I wanted to know like how are they progressing and where there is a niche where we can, from a formulation point of view, where we can fit in and help them to solve some of the problems they might be facing it. So I did some digging, and in one of the reviews, I published various clinical trials on different types of HSP90 inhibitors that were explored in National Cancer Institute as well as world over. I came to know there were two drug molecules like 17-AAG that was discovered by National Cancer Institute, was in phase 3 clinical trial, and NVP AUI 922, which is luminesipib, discovered in Cancer Research Institute, London. It was in phase 2 trial. So these were the two most explored HSP90 inhibitors in advanced clinical trials. So 17-AG, we also explored back in IISc, Indian Institute of Sciences. So I had like a pretty decent understanding of the chemistry of this molecule. The problem people that were facing in the clinical trial was that most of the trials used formulations with cremophore or DMSO, 1% or 2% DMSO in saline. So basically, if you dissolve your drug in 1 or 2% DMSO and compound it in a IV bag, it forms a nanodispersion. So that prompted the question in my head, like, why would you do a DMSO dispersion when there is an option of developing nanoparticles with a polymeric system attached to it? And that way you can actually help in having a better release kinetics as a control delivery system. So we discussed this options in one of our reviews where we discussed like HSP90 targeted inhibitors in nano anti-cancer therapy. So the figure which I've shown over here is the same thing which I was telling, that HSP90 is essential for carrying out various cellular functions associated with the cell survival in cancer. And as you know, like the cancer, as I told you before, that cancer has acidic pH. It's a hypoxic condition. There is a lot of turbulence in the cellular microenvironment. So it releases a transcription factor, which is called as heat shock factor 1, which is responsible for the production of this protein HSP90. So the idea was that just kill the HSP90 and the cell would die, but cancer is a smart disease. You kill it by killing one mechanism. There is always some backup that exists which prevents the total destruction of the cancer cell, and it just flourishes again. So there was this paper which suggested that there is a backup mechanism supported by mTOR, which reactivates heat shock factor 1 by its phosphorylation with, and mTOR is a kinase, so it causes a post-translational modification of the serine residues and and activates HSF1 for the production of HSP90. So no matter if you kill a cancer cell with only mTOR by giving an only mTOR inhibitor, or if you give only HSP90 inhibitor, the cells would still survive and it will still flourish. So we proposed that it will be possible, it will be an interesting concept if we have a dual drug loaded polymeric nanoparticles for the treatment of cancer. So now I'm moving towards from this, giving you a basic understanding of the heat shock protein 90 and how the nanotechnology can help in the delivery of heat shock protein 90 inhibitors. Now I'm slowly moving towards the concepts of the biomaterials. Like I was introduced to this whole new area of science where the materials were used and the chemistries were manipulated to create smart drug delivery carriers. So there are two basic strategies that is used in nanotechnology and drug delivery. That is, first one is the untargeted polymeric nanoformulation, and the second one is the targeted polymeric nanoformulation. So as the name suggests, it is untargeted. That means if you create a drug delivery carrier using polymers, these polymer drug nanoparticles would go and get absorbed by the tumor cells or the tumor tissue by a passive diffusion pathway. That means there is a chance that the nanoparticles would also be getting absorbed by normal cells, causing side effects. So some researchers suggested that what if we can coat these nanoparticles with some targeted moieties? That way, it can sense the nanoparticles. It can sense the cancer cells, and the cancer cells would be selectively taking majority of these nanoparticles. So that gave rise to the concept of targeted polymeric nanoformulations. So the targeted nanopolymeric formulations used targeting moieties like aptamers, folic acids, antibodies, um, even the small peptides, which can sense to the cell membrane surface proteins, for which are overexpressed in certain type of cancer cells, and that way it can actually go only towards selectively towards the cancer cell. That's what the idea was. So, to develop nanoparticle drug delivery for the 17AG, the classical approach would be to first create the chemistry of the polymers with the drug, because we did not know at that time how the drugs like 17AG or NVP AUY 922 would interact with the polymer. So we first thought of developing an untargeted nanoparticle and then go and move further in get developing more targeted approach. So we dig more into the literature and saw like this concept was already being developed by other researchers across the world. So I would like to mention, so this is like a history of the nanomedicinal history of 17AG, which is an HSP90 inhibitor being used and formulated in different types of nanoparticles for drug delivery applications, or like last 1 or 2 decades. So in 2009, this paper was published where a researcher from a Japanese professor, he or she actually used geldenomycin and magnetic hyperthermia in synergy for treatment of cancer. So now I'm introducing a term called as magnetic hyperthermia. What it means is that, like, when you give magnetic nanoparticles like Fe3O4 and expose it to an alternating magnetic field, such as in like just imagine an instrument like MRI, where you are exposing your body to an alternating magnetic field. Similarly, if you have a coil which can create an alternating magnetic field, and you put a magnetic nanoparticles in between, and you generate an electric if you generate a magnetic field around these magnetic particles, they have a tendency to resonate and cause localized heating. The heating is so dramatic that, like, if you take a solution of a magnetic nanoparticle and expose this to an alternating magnetic field, it can shoot the temperature from 0 degrees Celsius to approximately 60 degrees Celsius within 2 or 3 minutes, or around 5 minutes, depending upon the strength of the instrument or the magnetic field that you're using. But roughly around 60 degrees Celsius you will easily get in the low magnetic field area. So this in 2009, it was suggested that geldenomycin, which is a parent molecule to 17AG, was used in synergy with magnetic hyperthermia. I would like you to remember this analogy because this is the foundation of what we did later on. Then this in 2009 and 10, some people developed 17AG polymeric nanomyceles. In 2011-12, they developed rapamycin, mTOR, 17AG paclitaxel triple drug loaded polymeric nanoparticles. Then in 2012 and 13, they developed a targeted cancer therapy. So in 2013 and 15, we thought that why don't we combine the concept of magnetic hyperthermia along with 17AG, which is a second generation HSP90 inhibitor, and combine it in the polymeric shell. That's what we were trying to develop in 2013 and 15, and that's what I'm going to discuss now on. So the first formulation which we developed was 17AG loaded PLGA magnetic nanoparticles. The design was very simple. So you see a maroon color powder, when you it's when you put it near the magnet, it's just sticking to the magnet. So the maroon color comes from the drug because it's colored compound, and Fe3O4 is slightly darkish black colored crystalline powder. So when you create a polymeric nanocomposite and you expose it to the magnet, it sticks to it and it attracts towards it. This is like a raw or a layman way of understanding whether the particles that we synthesized was having a magnetic component in it or not. And these powdered particles, when you put it in the aqueous phase, it gives a uniform aqueous solution that you can see it on the screen. And when you again expose it to the magnetic field, these particles can get attracted towards the magnet. The idea was that, like, if you give these nanoparticles and treat cells, the cancer cells with it, it can give HSP90 mediated anti-cancer effect in combination with um, magnetic hyperthermia. When you expose these nanoparticles treated cells to an alternating magnetic field, please understand it's a magnetic field. So we are actually applying a magnetic field around these nanoparticles, making it to heat. So these two things of like chemotherapy combined with magnetic hyperthermia provided us a synergistic pancreatic cancer therapy. So now I'll be talking more about the characterizations of the 17 AAG in magnetic hyperthermia. So as you see over on the screen, I have just provided a short schematic of the synthesis where we used oil in water emulsion technique and sonication and simple evaporation to create nanoparticles. Although it looks simple, but the optimizations and creation of these nanoparticles took a lot of time and effort. But this is the simplest way that I can explain to you in this short period of time. So the nanoparticles, I created two different types of formulation. One is to 1:20 and one is to 1:10. And the Z average particle size were is I've shown it on the screen. It is 241 and 204. Now the table which I've shown, these were the experiments from the dynamic light scattering. So these dynamic light scattering experiments can be sometimes be difficult for reviewers to understand. So we confirm these experiments using SEM, that is scanning electron microscopy. The images are shown just next to it. So the first one is like a full bird eye view of the nanoparticles, where you can see the small spheres that you can see. And then we actually measured the particle size of each of these particles, which were close to around 200 nanometers. So this confirmed using scanning electron microscopy that we have synthesized nanoparticles. But now the question was whether they had drug in it, and as well as it has magnetic component in it, that is Fe3O4. So we need to characterize it using other techniques so that like we can answer these questions that we have. So before going into the characterization, I just want to give you some basic understanding of exactly what techniques we did and what are the basic principles behind it, so that like it's easy for me to explain you the results. The first one is electron scattering and characteristic X-rays. So I told you like these are nanoparticles, and then the first study that any nanotechnology person would do is to see the nanostructures under scanning electron microscopes or transmission electron microscopes. So this is the basic schematic of one of the technique that happens on its own along with the measurements or getting the images of electron microscopy. So as the method goes, in the electron microscopy, as the name suggests, you incident high voltage electron beam on the sample. And when you do that, you have like it shows on the screen that there is A and B, where A, it shows that the primary electron beam when hits to an atom. Now, I'm not talking from the perception of a nanoparticle, it's a big structure. I'm talking it from the perception of atomic. Now I'm going smaller. So think that you have a Fe3O4 atom which has K element shell, and you want to detect that this atom is actually belonging to Fe and nothing else. So you have a nanoparticle which has Fe in it, and you want, if you have incident an electron beam on the surface of these nanoparticles, what will happen is your primary electron will hit the second will knock out one of the electrons from these KLMN shells. So the example which is shown over here, it shows that it knocks out one of the electrons from the K shell, and it gets knocked out, and which is shown as a secondary electron. So this is one thing that can happen when you hit an electron beam on an atom. The other thing that can happen is the electron would just get deflected back, and it won't hit any of these electrons. So these electrons are called as backscattered electrons. Now, in case of SEM and TEMs, the secondary electrons are mostly used for getting the images. And there is also something called as backscattered electron microscopy that is used for something else. So when the secondary electrons are created, you see there is a void that is created. So C, D, and E shows you that there is a secondary electrons coming out. There is a void created in the figure D. So the arrow shows that the nearest uh, L shell has an electron which jumps into the K shell to fill up this void. As a result of this, to sustain this, to make it stable electronic structure, in the jumping of these electrons between the shell, it leads to an emission of photon or energy, and this energy or photon is called as X-rays or the characteristic X-rays. Please understand that these X-rays are the characteristic X-rays of the material, and they come out from a depth of approximately 2 micrometer, which is shown on the right hand side in the schematic. So the characteristic X-ray is a characteristic of a material. So if we can, when we are doing the transmission electron microscopy or scanning electron microscopy, the characteristic X-rays, invariably it comes out. And if you have a detector which is attached to this scanning electron microscope, you can actually detect these signals and confirm and verify that the material that you're looking at is having what elements in it, whether it has Fe, it has carbon, nitrogen, or other elements. Now you see the nanoparticles that I created had polymeric structure which had drug and Fe3O4. So it has a carbon composite. The carbon means it comes from, so the polymer consists of carbon, uh, maybe nitrogen. It doesn't have nitrogen because it's PLGA. So it has carbon, hydrogen, and oxygen atoms in it. And the drug has carbon, hydrogen, nitrogen, and oxygen in it. So it's mostly a carbon structure, organic structure, whereas Fe is an inorganic material and it's crystalline in nature. So to confirm the Fe presence in these nanoparticle, and our hypothesis was that these nanoparticle, when they self-assemble, the Fe particle would go inside the polymeric shell. So since it's present inside, the characteristic X-ray, which comes out from a depth of approximately 2 micrometer from a material, would be ideal confirmation that Fe is present inside the nanoparticles. So we performed some experiments regarding transmission electron microscopy, where unlike SEM, the transmission electron microscopy, the electron goes to the material. So to measure these characteristic X-rays, we performed transmission electron microscopy to see the internal structure of the nanoparticles that we synthesized. So you see over here on the left hand side, you have the nanoparticle structure, which is like a black color ball, and it doesn't have any contrast in it. Whereas on the right hand side, the two images that you see is a polymeric shell that is covering a gray colored contrasting small crystals. Now, we thought that these nanocrystals that you see, the dots, the black dots, contrasting dots, were nanoparticles encapsulated in the polymeric shell. Now, to confirm this, because we were doing transmission electron microscope and the electrons were constantly being fired at a high voltage, the X-rays characteristic X-rays were constantly being generated. And luckily, we had a detector to detect these X-rays, and voila, you can see over here in the graph at the bottom, you can see Fe Ka peak FeKb peak. So Ka is K alpha and K beta X rays, and Cu is coming from the background because these samples were like it was a nanodispersion that we put it on copper grid when we are doing a TEM measurement. So the Cu is coming from the background signal that is like a copper grid on which this nanoparticle is being placed. And Fe signals that came out confirmed that these crystals that you see in the images were coming from the Fe particles. This was a confirmation that we had Fe in it, but this was not confirming that we had drug in it. So we had other techniques that could actually say whether the drug is present in it or not. The simplest thing was that the visual observation that it gives a maroon color, so we knew that there was drug, but there has to be some spectroscopic quantitative method by which you can actually say that the drug is there in it. So we went to another technique which is called as X-ray photoelectron spectroscopy. Now this technique is different from the characteristic X-ray that I talked about. In this case, often some of my students get confused with this thing, and it's obvious. So if you Google this thing, you can easily differentiate between the characteristic X-ray and the XPS, the difference between these two techniques. But I'll try to explain this with the best possible in the best possible way. So what happens is that you have an X-ray bombarded directly on the sample. So when you bombard the X-rays, the electrons are ejected and the binding energy of these electrons are being measured. So you see the x-axis is the binding energy and y-axis is the intensity. So the binding energy of an electron for an element is a characteristic of that element. So this phenomenon of measurement of photoelectron spectroscopy was used for determining if there were Fe signals, if there were nitrogen signals coming in, and if there is carbon, oxygen, and other elements that were present in the nanoparticle powder that we synthesized. Now, as I told you, remember, like the nanoparticle polymer shell consists of drug and polymer. So the drug consists of nitrogen. So the nitrogen signal coming out from the polymeric nanocomposite is a signature for the drug that is present in the polymer. So the purple line that shows the Fe 304 MNP, these are the standard MNPs which doesn't have any polymer or any drug in it. Just to see that we can get Fe signals, these are standard. Then you have a blue color line which is a PLGA MNP 17 AG 1:1:20. So I told you that I made two polymeric ratios to see the impact of polymer on drug encapsulations and the release kinetic kind of thing. So the blue color is about 1:1:20. So 1 part of drug, 1 part of Fe3O4, and 20 parts of polymer. Similarly, 1 part of drug, 1 part of Fe3O4, and 10 parts of polymer. So these two formulations were studied using XPS, and the red line, which is at the bottom, is about the polymer, the drug, the standard drug. Now, if you see this whole scan spectra, it's very difficult to see a 400 peak over here, which I am just pointing the cursor to. It's extremely tiny. So we had to measure our, because these instruments are extremely sensitive and sometimes it's very difficult to measure the nitrogen signals from a molecule of a drug, because you need to think that right now you're thinking about a drug in terms of a single structure which consists of 1 or 2 nitrogen atoms. So from that perception, you have like at 400 a tiny bump. So we focused our the bottom spectra is like a full length scan, and the top spectra is like a narrow scan to see what's going on with the Fe, nitrogen, and carbon to understand the story of the polymeric covering. Now, the Fe overlay, it shows that the standard Fe is like there are 2 giant peaks that was confirming that Fe was there. But if you see the other 2 formulation, because of the polymeric cover, the signals were extremely small. On the other hand, because we confirmed Fe was present in using the transmission electron microscopy EDX, we were okay that maybe the polymeric shell is actually reducing the signals from Fe, which is coming from inside. The problem with Fe is with the XPS technique is that it is a surface phenomenon. Now, the characteristic X-rays comes out from within the material. XPS is a surface measurement. So it's measuring the signals that are existing from the molecules which are sticking on the surface of the molecule. So by chance, because we cannot control the self-assembly of the molecule, if the molecule is sticking outside, it's very easy to get a good signal. But Fe is within the shell, so it's very difficult to get a signal from the polymer. But we had like a small bump over here, and along with the TEM measurement and EDS, we were able to confirm that Fe is there. Whereas in case of nitrogen, you see the green color is coming from the 17 AG, which you can hardly see at around 400 binding keV, binding energy electron volts. So you can see over here, the green signal is coming from the nitrogen. The signal is slightly poor in case of 1:1:10, but it is almost not visible in case of 1:1:20. So we thought that this could be because of the polymeric shell, which is increased by 10 times, like it's increased by like twice. It's like it's 2 times. So we thought that because the polymer cover is so hard, it's very difficult to detect the nitrogen from the surface of the nanoparticles. So then what do we do? Like we had like a half cooked story that, yeah, there is a drug present in it and we can get the nitrogen. So we went to another technique for rescue. So you see, the classical formulation development would have uh, the drug release kinetics would actually solve the purpose, because if you do a UV visible spectroscopy, you can actually get a good signal of the drug present in, because the drug is actually a chromophore. So you can get a UV absorption maximum and you can create a linearity curve and release kinetics using it with along with your blank, of course. So using UV visible spectroscopy, XPS, we were able to confirm that the drug was present in it, and and using TEM XPS, we were able to confirm the Fe was also present in it. Now the release kinetics showed us that like the release was done. So that the two figures that are shown on the screen, it belongs to two formulations, that is 1:1:20 and 1:1:10. They were measured under two pH conditions, that is 7.4 and 4.5. The reason of having an acidic pH is because some studies suggested that the tumor microenvironment is acidic. It's approximately 4.5 to 5 pH. So we took 4.5 as a mark for checking our release kinetics. And what we found was that the release from 1:1:10 was slightly higher than that of 1:1:20, which is again an indication that the polymeric shell is driving the release kinetics. Further, we also map these release kinetics with some standard models in the world of the drug kinetics, like zero order, first order, Higuchi, which the table I've shown over here. So the data points were mapped for these release kinetic models. And what we found was that Higuchi and Korsmar papers had R square of like really high R squares in both acidic as well as pH condition for both the formulations. So we think that it was a Ficken it follows the Ficken law of diffusion and polymer erosion for the release of the drug. So in case of so what we think is that when you expose these polymeric nanoparticles in the aqueous phase for a long time, it swells because it absorbs water, it swells and diffuses the drug out of it. And because there is a pH variation, so in the acidic pH, it causes erosion of the pH and leads to the release of the drug and its pH dependent release that we could confirm using these experiments. So now the release is happening. We have a magnetic nanoparticles inside. So we thought of checking the strength of this magnetic nanoparticle. So what you see on the screen is it's the hysteresis curve that shows that the strength of the magnetic field for 1:1:20 is lower than that of 1:1:10 as expected, because the polymer is actually reducing the magnetic strength of the nanoparticles. So we thought that maybe this can also affect the biological outcome when we measure it for the hypothermia experiments. So we did the cell viability assays, and we found that the 17 AAG. So we did with two cell lines like MIAPACA2 and L929. I've not shown the data for L929 over here. I just wanted to focus on MIAPACA2 cell line, and we could see that like after 48 hours of incubation, we could get sufficient and like cytotoxicity for killing the cancer cells using drug PLGA MNP formulation for both the formulations. And since we were getting good cytotoxicities with MIAPACA 2 cell lines, we thought that like, okay, this is giving a cytotoxicity, but whether this will have a magnetic hyperthermia effect or not, because we have Fe3O4 present in it. So we went ahead with the magnetic hypothermia. So I would like to take some time to explain this setup to you. So this is not a coil that you use for, just want to tell you that this is not a coil that you use for heating up your water in winter. So I know Uttarakhand is extremely cold place, and even other places in the country we use the coil for heating up the water. But the coil that you see on the screen is generating a magnetic field. It's not heating the solution. It's creating a magnetic field. So if this magnetic field is on and if the buffer is running, if I put my finger inside, my fingers won't burn. But if that solution has magnetic particles in it, the magnetic particles would resonate in presence of these magnetic fields, and because of that it will heat up the solution to around 60 degrees Celsius, and that would be really problematic for me. It will burn my hand straight. So so the experimental setup that we had was a circulating water bath which was going through the tube, and you can see the 50 ml Falcon tube had three smaller Eppendorf tubes which were filled with the nanopart the cell suspension treated with drug loaded polymeric magnetic nanoparticles, and they were exposed to this magnetic field generator. And within 5 minutes, 5 or 10 minutes actually, we could actually see a reduction in the reduction in the viability of the cells. So the the untreated system was having the viability of around 100%, whereas both the treated ones reduced substantially, which was a confirmation that magnetic hyperthermia was killing the cells by generating localized heating inside the cell suspension, which were treated with these nanoparticles. So, having some experience with the drug delivery using the PLGA based systems, we went ahead. As I told you, like we were thinking of, we developed the rapamycin, which is an mTOR inhibitor, along with 17 AAG in PLGA, PLG loaded MNPs. So, it's a dual drug loaded PLG MNP system. So you can see in the video that I created, the nano you can have the nanoparticle powder which was lyophilized actually, and it's at room temperature right now. When you expose it to the magnetic field, the particles are so strong that a normal magnet with a normal magnet you can just drag the entire vial. And this thing is currently under development for further cytotoxic evaluations. But this drug, 17AG, was co-encapsulated with rapamycin in this system. And the screen shows the HPLC method, which was used for simultaneous estimation of 17AG and rapamycin in a single method. So the retention time for 17AG was 4.7 minutes. Rapamycin, it was 6.1 minutes. The SEM images confirm the nanoparticle size of these nanoparticles. The particle size distribution was around from 0 to 500 nanometers. And you can see the percentage presence of the drug was approximately 50%, which was confirmed using HPLC. So this work is still under development, and we are still working on this part. But this in for this experimental design gave us an idea that we could actually co-encapsulate three components in a PLGA polymeric system. So after having substantial experience with the PLGA, we went to another. We went. My lab actually worked on another system where we co-encapsulated curcumin and GAN61 in PLGA. The reason of developing this concept was that, like you see, like the cancer cells is like a heterogeneous population. So it was a concept which we discussed that like some of the cancer population in breast cancer, for example, in the tumor, there will be some population will be which will be a cancer stem cells, and there can be a normal tumor cells like cancer tumor cells. So cancer stem cells are highly dangerous because they are like resistant to some traditional chemotherapies that we give it for the treatment. GAN61 is known anti-cancer stem cell drug. So we co-encapsulated curcumin with GAN61 and in a PLGA polymeric shell. And the schematic shows the dual drug-loaded PLGA nanoparticle system, which could kill tumor cells as well as cancer stem cells. And what you can see on the right side is the same images of these nanoparticles, and these formulations were extensively characterized, and it was recently published. Now, having so you see, I started working with the PLGA, and then I went into curcumins, which is a natural material. So now I'm slowly and like entering into the world of something called as natural biomaterials for developing drug deliveries. So this particular formulation is already accepted and it's available in the market. It is called as Ebrexane. So this was my inspiration for developing albumin-based delivery systems. So albumin is like a natural protein which covers nearly 60% of the total blood protein. It's like a kind of a matrix which connects the cell. It's a natural drug delivery carrier which carries the drug between the tissues and cells. It it is extremely well characterized, and because it is a natural system, it's obvious like some researcher would actually use this chemistry for developing nanoparticles. So a researcher back in around 2005 or so 2001, around that period, they developed this ebrexane, which is a paclitaxel loaded albumin nanoparticles. And this formulation is still under clinical trials and helping developing multi it's being used in the clinical trials where multiple drugs are used for treatment of some cancers which are difficult to treat using monotherapy. So based on this concept, we thought that if it is possible, then we can actually develop uh, luminescipib, which is NVPAUI 922. So if you remember back, I showed you another drug, HSP90 inhibitor, which was developed in London, and it's a luminescipib BSA nanoparticle system. So as you can see on the slide, it is having the structure of luminescipib, the structure for albumin, and we used a simple desolvation method for creation of this nanoparticle solution. And this was used for treatment of MCF7 and MIAPAGA2 pancreatic cancer cell lines. The structure of these nanoparticles were confirmed using dynamic light scattering. And as you can see over here on the A panel shows the particle size distribution from 0 to 500 nanometer. The average particle size were approximately 220, if I'm correct. It's already there in the published literature. And the zeta potential was also negative. We confirmed the size of these nanoparticles using SEM and TEM. Now you see the TEM of this thing is like black. It doesn't have those crystalline particles. This is the classical TEM image of drug-loaded nanoparticle system. And if you have an inorganic metal particles like Fe3O4, or if you have something like crystalline particles like gold, you can see a contrast happening in the TEM images. But here it's clean and clear. So we have a good nanoparticle system. We wanted to check the interaction of the drug with the albumin, so we use fluorescence quenching experiment. So in the literature, if you look at the literature where people are working with the drug interactions with the albumin, people have been investigating the interaction of the drug because you see in the bound and the free form of the drug. Bound means bound to the albumin, and the free is like available for the pharmacological response. So people have been investigating the interaction of the albumin with the drug. We use the same platform for checking our interaction, and we found that like if you because albumin is a protein and it has a tendency to excite at a specific wavelength, it gives an emission of approximately around 340 nanometer. So you see in the A panel, the standard albumin has a spectra, a blue spectra that can be seen. If you keep the concentration of albumin constant, and if you keep on increasing the concentration of the drug would keep on binding with the albumin protein, and as a result, there will be a quenching in the spectra that would happen because of the interaction of the drug. This was a confirmation that increasing the drug concentration, there was a decrease in the fluorescence of the albumin protein. So we thought that if the nanoparticle synthesis has been completed, the fluorescence would be extremely diminished. That was seen in figure C, where the drug BSA nanoparticle, the fluorescence was around 20 units, whereas the BSA nanoparticle was like the native BSA nanoparticle, which doesn't have any drug, has a huge spectra which can be seen in C. This was a confirmation that the drug is interacting with the BSA to form a nanoparticle. But we wanted to further confirm this using the UV visible spectroscopy. So the figure B, which you can see over here, the green spectra is the absorption maximum coming out from the NVP AUI 922, or it is also called as luminescent. And you can see the red line, the red spectra, which is from the drug BSA nanoparticle, and the blue spectra, which is a straight line, which doesn't have any drug in it. This was again a confirmation that the drug was present with the drug BSA nanoparticle when it interacts with the albumin. Now, since it was interacting with the albumin, it posed a question, where would it go and bind in this protein? So we performed a classical blind um docking study where we thought that the drug would go and bind to any place in the albumin structure. So we created a grid box of 90 by 60 by 100, which covered the entire structure of the protein. And we asked the software Autodock tool to dock this molecule and give us the Gibbs free binding energy for various structures that can come out. And we found that this conformation, where it's shown in the model, is the most stable one with the Gibbs free energy of minus 10.48 kilocalorie per mole. And the interacting residues of each of these amino acid in this binding model is shown in the secondary structure, the structure over here. It's a two-dimensional structure, and and in the bottom there is a table which has like hydrogen bond interaction, polar interaction, cation pi, hydrophobic interactions that are happening between this luminescent and the albumin is also being shown along with its respective Gibbs free energy. So you see, this is the stable conformation. This is how you can visualize when the drug would interact with the albumin, and then when you cause the precipitation or complete folding of this protein, it would form a nanoparticle. So this is the time-dependent anti-cancer efficacy for the nanoparticles. I've not shown the drug release data, but it again shows the time-dependent drug release. It's already there online, so you can look at it if you are interested in it. But I just want to point out that like this nanoparticle was, you see over here, DNP, that is like drug-loaded nanoparticle for MCF7 and MIAPACA2 after 48 hours of time interval, was giving significant reduction in the cell viability after 48 hours. And because this is a LMR blue assay where we used UV visible plate readers for actually checking the cell viability, I wanted to see it visibly. Like I wanted to see like exactly how the morphology of the cell would look like when you actually treat these cells with the nanoparticles. So on the right side, I've just shown you an example of the MCF7 cell line treated with the native BSA nanoparticles. So you see the colony formation that you can clearly see for the MCF7 cell lines, which are sticking at the bottom of the T flask. And these cells are growing really well. And on the bottom, when you have the luminescent treated BSA cell lines, the cells were getting shrinked and the morphology was getting circular as if the cells were dying. And that was confirming our spectroscopic cell viability observations that nanoparticles were actually having a good anti-cancer efficacy against both the cell lines. Like MCF7 is shown, but it was also good against MIAPACA2 cell line. Now, albumin is an interesting like concept. So we also went into a lipid because, as you see, like in the first slide, I told you like I'll take an example from all the facet. So we covered a lipid base like PLGA. Then we had protein, that is albumin, and now again I am showing a lipid example where we used cholesterol and span 60 for delivery of another drug that was morosine. The nanoparticles were developed using thin film evaporation technique, and the particles were well characterized for its drug release kinetics, and it was also characterized using FTIR, and it was effective against the cancer. It was having a potential anti-cancer effect. And moving with this, then we have like a strong understanding of the biomaterials from the two important areas now, the lipid as well as the protein based system. And both the protein as well as the lipid based system were mostly used for killing the cancer cells, cytotoxic effects, and anti cancer therapy. But the next example that I'm going to show it to you it's an example of where we developed nanofibers for regenerative medicine. Here, this is an example of cellulose, which comes from a polysaccharide, and there are different types of polysaccharides that are available and being explored by various research groups. Cellulose is one of them. It's the cheapest source that was available, but we focused on bacterial cellulose from a reason that it was easy to grow the bacteria to get these cellulose, and then we actually sulfated these cellulose to create bacterial cellulose sulfate. And this bacterial cellulose sulfate was water soluble, whereas cellulose itself is non-water soluble. So we created water soluble bacterial cellulose sulfate, and in composite with PVA, we created nanofibers, which is shown in the figure. So you can see the top panel. These are the nanofibers that were created using PVA composites of bacterial cellulose sulfate. And we used electrospinning technology, where you create these solutions with various viscosities, and then you apply high voltage at the tip of the needle, where it will spray the solution and create nanofibers. So these nanofibers were later used to check the cytocompatibility as well as the adherence nature of L929 cell line, which is mice fibroblast cell line. And we can see over here on the bottom panel, you have the control, like the untreated sample. Then you have bacterial cellulose sulfate PVA solution only, and the nanofibers that were created. These you can see over here the blue and red staining which stains the DNA as well as the organelle of the healthy cells. You can see that the nanofibers actually helps to not only helps to grow the L929 cell, but also help them to stick on the surface, and the morphology was also healthy. So this was a unique like development that we did in terms of nanofibers, where we can actually show the use of these nanofibers for potential regenerative medicine applications. Now, Development of these nanofibers for regenerations and everything, you need a highly expensive technology or a tool which is called as electrospinning. If your lab has this technology, you can play with it. You can create different types of nanofibers at nanometer size. But I like to, but it's important to also know that you can still do science if you don't have anything. So this is the classical example where you can do fantastic science with the the least possible technological advancement that you can have it in your lab. Hydrogel, it's a well-known formulation that exists in the world of formulation science, and yet it is only known for its use for transdermal drug delivery. But now people are actually zooming in into the viscoelastic behavior of these formulations and using them as an injectable drug delivery system as an implant. So here on, I will show you some of the materials that we know that they were used for the nanoparticle synthesis, and you need some fancy tools like SEM or TEM for doing the characterization. The same material can be used for developing nanohydrogels. The only thing is, we need to go back to our basics of physical pharmacy, the textbook of Martin, where it talks about the classical concepts of Newtonian and non Newtonian system, the fluid nature or the viscoelastic behavior. So you see, when you have honey or any other material, like which is like fluidic and sticky, the first thing that comes to our head is the viscosity of the material. So the figures that I've shown over here is a classical system where when you do the rheology of these colloidal system, the viscous material, the viscous system can be classified as Newtonian or non-Newtonian, or it can also be further be classified as pseudoplastic, Newtonian, or dilatant system. Now, viscosity is a measure of ratio, it's a ratio of shear stress versus shear rate. So if you are doing a rheology, the viscosity would be simple. Shear stress divided by shear rate will give you the viscosity of a material. Now, it becomes little complicated when you talk about hydrogel, because hydrogels are not only viscous, they are viscoelastic in nature. So a Newtonian oil or a mineral oil would behave something like this. So this is a silicon oil. If I am doing a rheology of the system, the green line, it's the third axis, that is stress versus shear rate. It's an exponential line. That means as the shear rate increases, the shear stress also increases. It's a straight line. It's a Newtonian system. If you see the viscosity, it's a straight line. It's a Newtonian system. Whereas in case of hydrogel, it's a pseudoplastic system. So if you go back to your basic over here, the pseudoplastic system is a shear thinning system. So if you increase the shear rate, the viscosity reduces. And if you just check the stress versus shear rate, it's a pseudoplastic system, which is shown in the graph. Now, without going much into the technicalities and the details, we can, if you guys are interested in talking more about the rheology, you can email it to me in person. We can discuss about it. But in this short span, I just want to give you an idea, because we are moving into viscoelastic system. So you need to have some experiments to measure the viscosity along with its elasticity. So the rheology, this figure which I've shown over here, it's from a rheometer. This is a geometry. I'm showing it with the arrow, and you can see when you compress a gel and lift this geometry up, it creates a strings like this, which is called as a spin barkite behavior of a hydrogel. You see, this type of spin barkite behavior is only possible with the viscoelastic system, whereas the viscous system will be like, it won't create these type of stretchy behavior. So this stretchy behavior can be defined by some experiments. For that, I would like to define some or introduce you to some terms like storage modulus. which is a measure of elasticity of the material and ability of the material to store the energy. And the other one is the loss modulus, which is the ability of material to dissipate the energy. That means it is liquid. So in case of a hydrogel, the storage modulus is always higher than that of its loss modulus. This is how you can see whether a gel is there or not. So in both of these graphs, you can see the x-axis is your angular frequency, and y-axis is your storage and loss modulus. So to simplify, the blue line is a storage modulus which is higher than the loss modulus, means it's a hydrogel. And then in the other graph also you have a blue line higher than the loss modulus, then it is like a hydrogel. But you see, if you read more into this concepts of viscoelasticity and the formation of the hydrogel, some gels are physical in nature and some gels are chemical in nature. So you need some kind of physical force to form a gel, like heating, or you need some kind, or maybe pressure. So heating is the most common way of actually creation of a hydrogel, which is used for the biological application. Or you have like a chemically cross-linked systems like potassium ions, sodium ions, or maybe some kind of a cross-linker reaction, which can translate a liquid system instantaneously into hydrogel. So the physical cross-link hydrogel system have a classical signature where if you run a frequency sweep experiment, because this is an angular frequency on the x-axis, we term this experiment as a frequency sweep experiment. So if you run a frequency sweep experiment at any strain value, if you keep on increasing the strain or reducing the strain or anything, these two lines will never meet each other. First point. Second thing is, if you change the pH of the or the chemistry of this of these physically cross-linked system, these two lines, the frequency sweep experiments will not change. Whereas in chemical cross-linked systems, some of these systems are actually sensitive to the pH variation or the chemistry variations because they are cross-linked with some kind of a cross-linker. So you need some kind of a reaction to happen to tie the polymer pieces together to create a hydrogel. So you see, so over here, the storage modulus is slightly lower in the initial value of the frequency sweep. But as the angular frequency increases, the storage modulus keeps on increasing. That means that like when you are actually rotating your geometry on the stage, it's creating a shear, making the molecule come closer and closer and causing it to gel more and more. So there is a point at which these two things cross to each other, which some people refer to as the gelling point. But it's still, in the world of research, it's still a question that whether you call it as a gelling point or not. But some people do refer this as a gelling point. So, but from a perception of a physical or a chemically cross-linked system, this is how a frequency sweep experiment would tell you the gel that you have synthesized. Is it physical cross-linked gel or a chemically cross-linked gel? So the reason we went into the hydrogel is because you see some of the tumor forming cancers like breast cancers or the brain tumors, they are solid tumors and the tissue is soft in nature. So the classical approach to treat any solid tumor would be to surgically remove it and then provide a treatment with the chemotherapy or the radiation therapy uh, to remove the excess of the remaining cancer cell that might have been remaining in the cavity from where the tumor was removed. But there are also some implants that you can actually place in the cavity so that you can have a localized drug delivery. Problem is, we do not know how much viscosity, what should be the target viscosity of this implant, which is going to be in contact with the soft tissue. See, this implant has to come in contact with the soft tissue and it has to adapt to its microenvironment so that it doesn't cause inflammation and necrosis in the in the place in the tissue where it is being placed. The classical example for this is a glidal wafer. Glidal wafer is an implant, it's the only implant that we know of which is used for the treatment of glioblastoma. After the surgical removal of this brain tumor, glioblastoma, there is a cavity that is created in the brain. Once this cavity is created, surgeons like neurosurgeons would or the oncosurgeons would put these glidal wafers, which is like a size of a 1 rupee coin, into this cavity, and they will put a clip on this tissue and leave it there so that the drug can be released in the localized area. If you look at this formulation, it is full of problems such as impaired wound healing, cerebral edema, inflammations, and all the issues that are related to the formulation and not because of the drug. So we spoke to a lot of the some of my neurosurgery friends over here, and they said that this is a common problem, and we do not since we do not have any other option, this is the only thing that we can do for the treatment. So we diverted our attention towards the soft gels which are injectable in nature and we can place it in the cavity. But if we want to put it in the brain, what is the viscosity of the brain? How would this gel behave? How much hard gel I want to keep over there? So we performed some experiments over here to understand the viscoelastic behavior of the brain tissue. So the top panel that you see over here, it's the brain tissue of a mice, and it's a geometry. You compress it so you can see the brain tissue being crushed, and we ran two experiments with it where we did this frequency sweep experiment to see if the viscoelastic behavior of the gel, is it matching to a chemically cross-linked gel or is it matching to a physical cross-linked gel. So to our happiness, it was mostly towards the physically cross-linked gel system. And we were also able to see that from the stress versus shear rate curve, that the viscoelastic behavior of the brain is pseudoplastic in nature, which is common with most of the hydrogel system. Now, to our experience, most of the natural materials like polysaccharides like chitosan, carrageenan, alginates, hyaluronic acid, all these biomaterials have a tendency to form physically cross-linked hydrogel systems. But instead of using something which is from the plant source or the animal source, we thought of going for the human source. So we developed albumin hydrogels. Now, this is a classical example where we're building a albumin hydrogel. You remember we developed albumin nanoparticles. This is an example where where a single material can be used for either making nanoparticle, or it can also be used for making a clear, transparent hydrogels. If you manipulate the chemistry of this molecule, just think this way: you have an egg and you put it on a hot frying pan. You have a white albumin. It translates into something called as a half fry, which is like a white color blob. If we do some changes in the chemistry of this molecule, the denaturation in presence of the heat can be controlled and it can lead to a transparent gel system. That's what we did over here. So we made albumin solution in 25 millimolar sodium chloride solution, and we heated it in a controlled manner at for around 30 to 40 minutes at around 85 degrees Celsius. It translates into a transparent gel system that you can see over here. It can also form a gel with the water, but it's still confusing. But the interesting thing was this gel was a physical gel, as you can see from the graph here. And if we increase the concentration of the gel, of the material, it starts to form a structured gel. So we can actually control the thickness or the viscoelastic behavior of the gel based on its concentration. And since this gel is not affected because of the pH change, its structure, it doesn't change, we think that it is a physical gel system. We loaded drug to this gel, and we loaded doxorubicin in the albumin. We performed the rheology. We can see the frequency sweep experiment again confirms that the gel is physical in nature compared to the native albumin gel. And then we perform live dead assay, the cytotoxicity assay for the drug loaded albumin gel, where there was a substantial reduction in the cell viability of MCF7 cell line. You can see over here the background of this inverted microscope image, where you can see the background of the gel, and the dots that you see is like the cell dying because of the drug which is present in the gel. This is like 5,000 nanogram per ml, and this was like 1,000 nanogram per ml. The 1,000 nanogram per ml, you can see the cells are still alive, and it is for having the colony structures over here, but this it's still dying, but it's not efficient enough, which is going in line with the live cell assay that we did over here. So we have both visible observation as well as the cell cytotoxicity assay that could confirm that the albumin gels are interesting personalized delivery system, nanodrug delivery system. Now, I've been giving you an example of the physical system. Now, going to the chemically cross-linked system, where we used PVA boric acid reaction to form a cross-linking reaction over here, where we mixed it with the carrageenan. Now, carrageenan on its own forms a gel which is physical in nature, and so it doesn't cross. But we mixed PVA boric acid along with the carrageenan in specific ratios. So what happened was, when you have a solution or a viscous material of carrageenan mixed with PVA and boric acid at pH 4 to 4.5, it's fluidic, it flows down, it doesn't gel. But the moment you change the pH to 4.5 to 8.5, it instantaneously translates into a thick gel with the spin barkite behavior that you can see. And also, this physical behavior gets translated into a chemically cross-linked system. Now, this reaction of pH dependent change from physical to chemically cross-linked system is reversible in nature, and we are currently exploring this for drug delivery applications. Other than so, to summarize, we have developed a lot of drug delivery systems. We used albumins for developing hydrogels, nanoparticles. We use cholesterols for developing nanoparticles. We also use polysaccharides for developing gels, and so carrageenan is like a polysaccharide based gel. So we develop PVA boric acid carrageenan hydrogel for for the drug delivery. So we have been experimenting a lot with the chemistries of these biomaterials, the natural biomaterials, which can be used for personalized drug delivery applications. Other than that, the slide that I've shown over here, we are also so I'm also involved into translation aspect of my lab, where we do lot of we get a lot of samples from the clinical investigators in Jefferson Hospital. We perform clinical metabolomics as well as PK studies for various drug molecules, as well as the stability studies for the compounded formulations in Thomas Jefferson University Hospital. I have covered broadly what we are doing in our lab, but if you are interested to know more about the world of nanomaterials and drug deliveries and all those things, as Dr. Disha told, like I have published a lot of book chapters. So these are some important book chapters that you can read, and you can get acquainted to the world of nanomaterials and maybe experiment in your own labs, where you can develop better scientific formulations and solve some important problems in the field of drug deliveries, like targeted drug deliveries or tissue specific viscoelastic problems that we have with the current formulation designs. Other than that, I would like to mention that I am also guest editing a special issue in advances in polymers, which is a 3.4 impact journal, as you can see. The name of the issue is Advances in Natural or Synthetic Biomaterials for Developing Micro and Nano Drug Delivery Systems. The issue is open for accepting manuscripts, and it's indexed in PubMed, Web of Science, Scopus, as I told in the start of this session. Acknowledgement. This is my lab, and my mentor, my friend, Dr. Gagan Kaushal. Professor Alok Bhushan, Dr. Shakti Kumar. He was my mentor in Japan, and he's a good mentor. Dr. Nakajima, Dr. Toru Maikawa. The funding for all this work was funded partly by Bionano Group in Japan, from the mixed fellowship that I got for my PhD, InnoEndro grant, as well as RO1 funding from NIH in my current lab. This is just an information for some of the master students if they are interested. We have introduced Master in Pharmaceutical Sciences program. So if you are interested to get involved in the clinical projects like clinical trials and studying the pharmacokinetics, dynamics, LCMS related work, or you are interested into formulation designs, like get associated with the formulations, or you want to work on the molecular cellular levels like pharmacology of cancer cell biology, metabolomics, you can join our program in Master in Pharmaceutical Sciences. You can contact me or Dr. Or Professor Alok Bhushan. The name is there on the slide, or Christine Lord, who is the administrative assistant. And the interesting thing is, program is new, and it would be interesting for the budding pharmaceutical, the senior pharmacy graduates like B Farm graduates to join for the master's program. And I think it is giving a GRE waiver, but you can obviously check with Dr. Bhushan about this. And thank you so much for your attention. And I will be happy to take any questions that you may have. I hope you like the talk, and in the small span, I could squeeze as much as I can. Thank you so much.
Thank you so much, sir. It was outstanding and indeed very informative, and I hope it will be very beneficial for the students as well as the researchers watching us. So there is one question from Minakshi Pandey.
Okay.
She has asked, Sir, can you tell us some more newly discovered biomaterial used in nanotechnology?
Okay, so like there are lots of newly discovered biomaterials in the nanotechnology. So there are like PEG PLGA nanoparticles, and like these materials are already available. The classical biomaterials are already available for market use and Sigma Aldrich, and you can find lot in the market. But you see, like when you say biomaterial, there are like different types of biomaterials. Some materials are called as nanomaterials, like graphene or carbon nanotubes or fullerenes. These are carbon nanomaterials, which has more potential in the physical applications in developing various sensors or the biosensors. And then there are certain biomaterials which we use it for the formulations. So we, as a formulation scientist, we get inspired from the physics of the nanomaterials, and we try to use these biomaterials for our own applications. So there are like to answer your question straight, we have like pegylated systems. We have like polymethaacrylate biomaterials which are used for developing pH sensitive hydrogel systems, and we have lot of details that you can find. And the book chapter that I mentioned is discussing about all these biomaterials that I'm talking about.
Okay. Thank you, sir. There is one more question from Akhilesh Kotial. It is that could you be able to share the challenges and limitations of the formulation, if possible?
Okay, so like scientifically, so I will be more towards the scientific part instead of the regulatory part. Okay, so from the scientific perception, the biggest challenge is the encapsulation efficiencies of these nanoparticles. So the techniques that we did, the formulation development that we did, we were able to get like encapsulation efficiencies like around close to 30 or 40%, or maybe 50%. It would be interesting for the scientific community to think about the methods that can be used to have a better encapsulation efficiencies, so that we can minimize the wastage of the drug molecules. You see, the drug molecules for which we create a formulation itself is very expensive. You won't get these luminosepep and all those things cheap as aspirin or paracetamol. So wastage of these material with lesser encapsulation efficiencies can be a situation. So we our lab is more towards like developing better strategies to increase the encapsulation efficiencies. Another another place where I think it is a challenge is like the clinical translation part. You see, the dosing that is required to translate a formulation from a mice or a rat model into a human, it's just unimaginable. How would you decide how much dose you are going to give in a phase 1 clinical trial? If you are giving a mice which is like around like 200-gram animal, imagine a 60 kg human, you want to translate this formulation into a human use. So if I want to ask my clinical fellows over here, "Okay, please, I have this powder and I want to do a translation of this thing," they will be in a question, "Hey Ankit, how much should we give? Where should we start from?" So these questions are always challenging in the world of translational aspect, and would be interesting to have better models to answer these questions.
Okay, well said. And sir, there is one more question from Suma ma'am. As per your studies, which is the best target, lipid or protein for cancer therapy?
uh for cancer therapy as such, people focus mostly on proteins because proteins are like drivers of the cellular machinery. So you see, the lipids, it only forms the cell membrane. It just forms a covering of an organelle or a cell. Okay, it's like a cover of everything. But what goes inside, which drives that cell, is going to be the target. So mostly proteins are considered as a drug target for cancer therapy. But from formulation point of view, I prefer to go for proteins like a bulk material. See, you can actually extract albumin in grams from a bovine source. Okay. But if I ask you to extract DNA in grams, will that be possible? In current scenario, it's a technical challenge, but there are labs who have reported DNA gels. In the world of biology, people use DNA as a molecule for cell function or a genetic function. But for a material scientist or a formulation scientist, it's a bulk material which is gelatinous in nature and you can make a gel out of it. So you see, from the two perception, from the cell biology, it's the protein that is responsible for carrying out the cellular function. From a formulation point of view, it's always better to look for the protein as a drug delivery because it's like abundantly available. Whereas the polysaccharide, we don't have like we our body is full of monosaccharides and disaccharides. We don't have polysaccharides floating in our body. So you can't really extract a polysaccharide from human body. I mean, human cannot synthesize starch or cellulose, which is used by a formulation scientist for creating a formulation. But you can extract proteins like albumin. You can extract and scale up the extraction of a DNA to create a bulk material which can be used for the formulation. So from both the side, you need to decide what you're looking for. I hope this answers.
Okay, thank you very much, sir. Now I welcome Dr. Varsha Tiwari, Professor DVCP, for the vote of thanks. Welcome, ma'am.
Thank you, ma'am. Thank you very much, sir. It was an excellent session. We have completely understood about the anti-cancer therapy, HSP90 protein inhibitors, along with mechanism of action, formulation, development of nanoparticles, rationale, procedure by using magnetic hyperthermia, proving the nanoparticles, and also proving the nanoparticles possess drug as well as its chemistry. We also came to know the new technique, X-ray photoelectron spectroscopy, how we prove the drug is placed within the nanoparticles, and also about the hydrogens. It was purely research-based session. In the end, I want to say in Hindi, you would like, I think, आपने कागज में कागज भर दिया. It was excellent session. Thank you very much.
Thank you so much, ma'am. But I know it was too much of science and too much of research-oriented activity. I wanted to tell all of you in the beginning, but I think it skipped my mind because it's science, So I just keep on talking about it.
No,
Sorry.
no, no, sir. It was very good. Thank you. Thank you very
Thank you
much.
so much.
Thank you, sir, and thank you, ma'am.
Thank you, ma'am. Now, on behalf of Devsthali Vidyapeeth College of Pharmacy, presenting you a token of love for your kindly accept it, sir.
Thank you. Thank you so much. So much.
Thank you once again. Now, it's time to take a break, but we will come back after 1 p.m. So stay tuned for all the viewers. Be on time. We will take a lunch break and we'll come right back at 1 p.m. Thank you.
Good afternoon, sir.
आवाज नहीं आ रही।
Hello.
आवाज नहीं आ रही। आप बोलो आवाज।
Hello. Good afternoon, sir. Am I audible?
Yeah.
Good afternoon. Good afternoon.
Good afternoon. Very good afternoon.
Yeah,
To
good
one
afternoon, sir.
here to attend the second session of fourth day of PFC National Conference. Today, the speaker is Dr. Anand A. Mahajan, Associate Professor, Goa College of Pharmacy. His today's topic of discussion is application of LCMS MS for characterization of degradation products. I request all the delegates and participants to enjoy the session. Now I would like to invite Ms. Disha to give the brief introduction about the speaker. Thank you.
Thank you, sir. Good afternoon, all in one. Our upcoming session. Good afternoon, everyone. Our upcoming session is going to be delivered by Dr. Anand A. Mahajan on the topic applications of LC-MS for characterization of degradation products. Dr. Mahajan is a well-known name in the field of education and research and is presently working as Associate Professor in Pharmaceutical Analysis in Goa College of Pharmacy, Panaji. He is having more than 18 years of experience. He did his doctoral studies from NMIMS University, Mumbai, and graduation and post-graduation from Pune University. He had more than 12 international and more than 20 national publications in highly ranked peer-reviewed journals. He had done many projects as principal investigator funded by various funding agencies like University of Pune, Generic Healthcare Private Limited Pune, Prerna Fine Chemicals, Aventis Pharma, Shogun Organics, and so on. He won various awards and recognitions, won second prize for scientific poster presentation for poster titled Formulation Development of Low Solubility Drug Substance by Solid Dispersion Technique conducted by Innopharma in October 2018, Panji. He was awarded for Dr. P.D. Sethi Annual Award for 3 times for his research work in title like LCMS studies for the identification and characterization of degradation products of hydrochlorothiazide and establish of mechanistic approach towards degradation in 2012. Then the development and validation of high performance thin layer chromatographic method for estimation of brimonidine tartrate as bulk drug and in ophthalmic solutions in 2010 and validated high performance thin layer chromatographic method for estimation of olopatadine hydrochloride as bulk drug and in ophthalmic solutions in 2010. He also won Best Faculty Award of SSP School of Pharmacy and Technology Management, NMIMS University, Mumbai in the year 2009. He also got the Best Suggestion Award by Vocard Life Sciences Limited, Aurangabad in the year 1999 and 2000. His work has been presented in different national and international conferences and seminars. He had also chaired different scientific sessions in national and international seminar and conferences. He had also attended various faculty development programs. So without further delay, I heartily welcome Dr. Anand Ashish Mahajan. Welcome, sir, and the stage is all yours. Sir, please continue.
Am I audible?
Yes, sir, you are.
Okay. So, before starting of the session, I would like to express my thanks to the patrons, trustees of Devsthali Vidyapeeth, Rudrapur, Uttarakhand, for their support to the 5 days national conference. And I am also thankful to Power Finance Corporation, New Delhi, for sponsoring the same. Special thanks to Principal Sir Dr. Ashok Kumar for his cordial invitation, because of which I am able to share this common platform with you all. I am also thankful to organizing committee and faculty Devsthali Vidyapeeth, students who volunteered for the same, for their efforts in making this program a great success. I am also attending this program from last 3 days. A word of appreciation to all students who are aspiring knowledge and learning new things out of this conference. And I'm also thankful to Disha Datta Madam for my introduction. So now I would like to uh, do or express my presentation. The outcome of this study is, after this presentation, students and faculty can take up academic research with some novelty. And because of that, your work gets published into a good impact factor journey. So I will take you through a good approach that how you can take up your work, okay, from laboratory to a good journal, and you should receive some amount of appreciation from the faculties or from the researchers who are doing work into that area. So I would like to share my slides. Now, can it be possible, ma'am, to share my slides on the screen?
Yeah, yes, sir, definitely.
Yeah. So can I start now?
Yes, sir, please continue.
Yeah. Thank you. See here, I would like to tell you about a very important application of LC and LCMS in characterization of degradation products. Now, content of the presentation is, first I would like to tell you about regulatory aspect, why it is important. Why? Because most of the pharma industries are doing work for their regulatory queries or regulatory requirements. Then I'll tell you that how drug candidates is to be selected for such type of study, and what are the criteria for a student or teacher to select a drug candidate. Third thing is, I'll tell you that how chromatographic system is, okay, or how chromatographic system is selected, and how you should shift your method from liquid chromatography to mass spectrometry. Then I have put two case studies and go through case study one, from where I learned a lot of things. Okay, so if you also share this case study with me, okay, it will be very good experience for all of us. And then I'll go through case study two. So here, what is regulatory aspect? So we will be shifting to regulatory aspect slide. So this earlier registration and documentation was country specific. There is no uniformity. Okay, if you want to register a product, maybe API or formulation, USA or US FDA having their own guidelines. Okay, EDQM, EMEA in Europe, they are having their own guidelines. Ministry of Health and Welfare, Japan. So these three countries, they are having their own individual guidelines. Suppose I am sitting in India, I want to manufacture a product, and want to register in USA, Europe or Japan, I have to follow their guidelines, which were different from each other. So it was very hectic to put a product for a stability condition which is mentioned by USA. Then same product we have to look upon what European guideline says. Then same product we have to again look upon what Japanese guideline is stating. But what happened in 1990, we will go to the next slide, and all of you will come to know that in 1990, there is a constitution of ICH Committee, International Conference on Harmonization. So what ICH did, okay, people from Europe, USA, Japan, along with the experts from pharmaceutical country, this ICH, okay, called them together and formulated a new guideline. And that's why ICH guidelines are important. Most of the ICH guidelines is being taken by most of the pharmacopoeias. So now it becomes a legal binding. So whenever you carry out any studies, people will prefer to follow ICH guidelines because most of these guidelines are common with respect to USA, Europe and Japan, which is a big market for Indian pharmaceuticals. Now we will go through a quick glance through various ICH guidelines and then you will understand what is the importance of this degradation characterization. So this is an ICH guideline Q1AR2 related to stability testing of new drug substance and products. And you know that stability issue is one of the most important issues into pharmaceutical product manufacture, storage and selling. So if you see this under point 2, okay, this guideline shows that stress testing is one of the integral part of the what your study. You should carry out stress testing of your formulation. You should see that whether your product is stable or susceptible for degradation. If the product is susceptible for degradation, ICH had set identification threshold as well as qualification threshold. If your product or any impurity crosses that, then you have to identify if it crosses identification threshold, and you need to qualify, means toxicity profiling of that impurity has to be carried out. But not only this guideline, there are other guidelines also which explicitly required to carry out stress studies and characterization of degradation product. We'll look upon these guidelines as this is a regulatory aspect. Everyone should understand why industry is doing this work. Q1B is a photostability testing guideline put forth by ICH. What they are saying? Forced degradation methods are designed to provide suitable information to develop and validate test methods. So again, they are telling that you need to carry out forced degradation methods so that you will understand the nature of your formulation and stability aspect of your formulation. Then there is a Q2R1 guideline. It is related to validation of analytical procedures, text and methodology. What it says, as appropriate, this should include samples stored under relevant stress conditions, light, heat, humidity, acid oblique base hydrolysis, and oxidation. So what they are telling, you expose your product to all these conditions, which product naturally experience when it is on shelf and after manufacturing, and see to it that what is the stability of your product. Then there is another guideline, Q3AR2, impurities in new drug substance. So what it is saying, the registration application should include documented evidence that analytical procedures are validated and suitable for detection and quantification of impurities. And you know that ICH has classified degradation product as one of the impurities. Another guideline Q3B, impurities in new drug products. The registration application should include documented evidence that the analytical procedures are validated and suitable for detection and quantification of degradation products. So not only one, but if you see this, stability issues is being addressed by lots of ICH guidelines. And you know that nowadays, ICH guidelines is one of the most important guidelines which satisfies many regulatory conditions as it is being formed by three countries' guidelines came together and they formulated a single guideline. So Q7A GMP guideline, what it is telling, the test procedures used in stability system should be validated and stability indicated. So I told you that these guidelines are incorporated into official book like USP. These are the chapters where this issue is being addressed. 1150, 1191. So what 1150 says, monograph assay method should be used for stability testing if they are stability indicated. So you need to check these monograph methods where they can use it for stability studies. 1191 chapter, at appropriate time interval, sample of the product are assayed for potency by use of stability indicated method. So once it is included into official book, it becomes a legal binder. Now FDA, earlier FDA has formulated this guideline for new drug substance and new drug products, but later on the same guidelines is being okay, put forth for abbreviated new drug applications which were there for impurities in new drug substance and new drug product. So FDA wants the safety issue should be equally important with the generic drug as well as it is important for new drug substance and formulation. Now, why you required to carry out force degradation? What are the various aspects? Okay, you are important in carrying out force degradation study. So, first is intrinsic stability. It will tell you about how much stable is your formulation as well as your drug. So the stability studies are being carried out for APIs as well as for formulation. So this will tell you about intrinsic stability. So you'll come to know how much my formulation and API is stable. Second, ICH tells degradation pathway enables pharmacists to design robust formulation. So when you study degradation pathways, I'll tell you that how we predict degradation pathways, prediction. Okay, I try to cover all the aspects so that you can take up it as a project at your college level and can do something and publish in a good journal. So degradation pathway will tell you that whether your product is light sensitive. If light sensitive is there, then you need to have a coating over tablet. This will tell you that whether your product is susceptible for oxidation. If it is susceptible for oxidation, you need to take care that minimum exposure of that drug should be there. There are some products which are prone for hydrolysis. So whenever they came into contact with moisture, a little bit acid or alkali into the formulation, they undergo hydrolysis and they get degraded. So these degradation pathways are very much important for a formulator as well as analytical chemist to establish a robust formulation. Now, third most important thing is it fulfills regulatory demands for impurities in both new drug substance and new drug application. And as I told you, it is now equally applicable to ANDAs if you file with your generic formulation. Now, then fourth thing, it determines qualification threshold of impurities which gives safety of these impurities. Now, if you slowly have seen progress, earlier all methods were assay methods, and we used to do assay using UV spectrophotometry or potentiometry. Now slowly, because of this impurity issues, all the method is being replaced by HPLC methods. And cost is the only one factor. If the cost of LCMS if is affordable, then I'm guessing that in next 10 to 20 years, all monograph methods will be replaced by LCMS methods. LCMS methods, they are very much accurate in identification and quantification of impurities. Very less time is required for method development, as you can identify impurity depending upon its mass. And I think in next 10 years, okay, all monograph method is being replaced by LCMS methods. Another important thing and commercial point of view, impurity reference standards have huge demand in the market. You can see the cost of one impurity sample. Okay, there are certain impurities which are so costly that even at college level we can afford it to purchase those impurities and carry out study. So it has formulation importance, it has regulatory importance, it has safety concern, and it has commercial value. So because of this activity becomes almost important for any of the pharmacists, for any of the analytical chemists, as well as formulator. Now, most important thing is selection of the drug. See, most of the time what happens is in academic research, as we do not have that strong commercial platform where we can demand today and can purchase tomorrow. So most of the time what we do is student okay, sees a molecule, look upon literature and sees there is no novel method reported and immediately what? Write application letter for a gift sample and get the sample. But when sample comes, okay, he or she observes that there is no UV absorbance for that drug. So almost you have written an application, you have okay, wasted your 15 days to receive sample, and once you receive, you have only HPLC system having UV detector, and your sample do not have that much strong UV absorbance. Then you cannot carry out a research further. Otherwise, what happens? You import a drug from abroad also, from here also you receive gift sample, you carry out your study, and before you publish, somebody else has already published that drug. So these are the lacunas in academic research. So what I will tell you is, for this type of work, do not take same type of nucleus. Why? Because 80% of the study is already reported into the literature. You are not doing anything novel. If you see a major nucleus of amlodipine, felodipine, nicardipine, so major nucleus is the same. There is a difference in side chain. So there is no point in taking another dipine for consideration. Why? Because you are not doing anything novel. You will not learn any new chemistry involved into degradation, into establishment of degradation products, and looking after the toxicity of that degradation product. Why? Because the paper amlodipine, we only published amlodipine paper where we have almost characterized and separated 7 degradation products. Okay, if there is a candidate who is taking felodipine, I think few of the degradation product will be same like amlodipine, and you cannot achieve anything novel. Same like proton pump inhibitor, rabiprazole, pantoprazole, lansoprazole. See, major nucleus is the same if you see that. So for such type of study, do not take up a nucleus for which already there have been lot of paper published. So you have to take variety of the chemical scaffold for this type of the study. If you consider variety of the chemical scaffold, then you will establish new degradation pathways. Then you will come out with something new, and then your paper will get cited and requested by other authors. So if you see this, good variation of nucleus is found in following class. If you see antiviral drug, dolutegravir, a totally different nucleus is there. Telaviridine, you have a completely different nucleus. Darunavir, you have completely different nucleus. Okay, so my objective of taking up research that I should take a different chemical scaffold which is not related to their previous okay drug molecules. Recently, I came across with one lecture, and then I came to know that darunavir is being discovered by one of the Indian scientists in the United States, and his name is Arun K. Ghosh. I got literally stunned that an Indian scientist has discovered such a wonderful molecule. Arun K. Ghosh belongs to Bengal, and when you put into Google Darun in Bengali, the meaning is great. So that's why the name he kept for this molecule called as darunavir. Even you can put his name into the Google. You can search. Okay, you can get a glimpse of his laboratories into United States and how they are carrying out drug discoveries. So antihyperlipidemic, if you see ezetimibe, different chemical scaffold, simvastatin, different chemical scaffold, atorvastatin, different. So I will encourage all of you and faculty to take up a different nucleus if you would like to carry out such type of work. So you will definitely come out a new type of chemistry which is involved in degradation products and characterization. Now, next we'll be looking at basic things, how we can select a chromatographic system. So I did a lot of PhD thesis, a lot of paper for evaluation, but when you study or read results and discussion, they have included or they include a very poor result and discussion. What they included, I have increased micron concentration from 30 to 40%. Then there is a shift of retention time. Then pH is adjusted to 3. Then it is adjusted to 3.5. Then there is a retention time. But there is nowhere, okay, student or researcher discuss about a chemistry or logic involved into chromatographic method development. Okay, so here we will learn how we will put lot of chemistry into that, how we will write a result discussion part into that, okay, which will increase definitely a good weightage for our publication as well as our thesis and a research work. So HPLC system, any system you can use for such type of work, even if it is binary, will be okay. Quaternary, no problem. Illusion, preferably isocratic illusion, you do initially if you are not very well versed with such type of study. But majority of cases what happens, you get polar degradant and non-polar degradants forming degradation study. In that case, isocratic illusion fail to resolve this degradation part. So any binary system, manual injector, even if it has, will be fine. For academic research, I will only suggest that if you do not have a very high funding, please go to a binary basic system, okay, which have manual injector, okay, without column oven, right, preliminary. Why? Because maintenance cost is low. If you go to quaternary system, auto sampler, even if there is a damage to one sensor, okay, the whole system will be for under maintenance. We do have binary system also, quaternary system also, but our binary manual injector is absolutely doing well. On this quaternary, okay, hi-fi systems are frequently under maintenance because they do have a lot of sensors. If it is have auto sampler, again it will have sensor. There is a needle problem, air into the needle. So I will advise if you would like to carry out an academic, even you want to publish a good paper, a binary system with manual injector and a diode array detector will be preferred. Even your budget do not permit diode array detector, UV detector with variable wavelength selection is also absolutely fine. You will find that there is a huge cost difference between diode array detector and UV detector. But no doubt, diode array detector will tell you a very informative things about a degradation product, eat purity things, but if you cannot afford, still you can carry out a good work with an UV detector, which is having a variable wavelength selection facility. Buffers, see definitely for characterization, you need to shift your method from HPLC to LCMS. So you require a volatile buffer because you know that your ion should be in a gaseous state when you analyze by your LC-MS. So you need a volatile buffer so that this buffer should not deposit any salt material. If I take potassium hydrogen phosphate, which is not a volatile buffer, then I will get a lot of salt deposit into my MS system. And the performance of MS system will not be good if we use non-volatile buffer. So most frequently used buffer for LCMS systems are ammonium formate and ammonium acetate. You can adjust ammonium formate buffer pH using formic acid and ammonium acetate you can adjust with acetic acid. Now most important thing is most of the students don't write resultant discussion why they have selected a pH of their mobile phase plus and minus 2 ppm. Most of the cases I found that they start with 2.5 and they go after change of 1 pH and they carry out separately 2.5, 3.5, 4.5, 5.5. If you ask them why you have chosen all these pH for your study, so the answer is to look upon a retention time of the drug, how it gets changed. So see, in next slide, I will tell you the basic why it is recommended that pH of the mobile phase has to be selected to plus and minus 2 value of EQA. So we will study next slides and there we will understand what is the role of pH in design electronics. So before that, we will learn couple of things related to mass, though these are the basic things. But what happens is generally, if I want to carry out an hi-fi research, I generally forget basic things. But basic things are only basis for the development of method and characterization of these methods. Okay. For baseline stabilization, generally a lot of students has a problem in baseline stabilization. Why? Because they have a problem in baseline stabilization. Most of the time they use C18 column and you know C18 column is non-polar in nature. So if you start a mobile phase having a ratio with high quantity of buffer and less quantity of either methanol or acetonitrile. See, if you start with high quantity of buffer and your stationary phase is non-polar and you are getting mobile phase highly polar, there is a lot of time your column will take in baseline stabilization. So what I will advise you, here is the table. Okay. So organic phase generally you start with 90 because we work mostly on C18 column, which is non-polar. And if you take organic phase in higher concentration, there is a good compatibility and you get a very fast baseline stabilization. Many times I see that students are keeping column stabilization for almost 1 hour when I see that they use mobile phase 95% buffer, 5% buffer. See, you have to use such a logic. C18 column, non-polar. For that non-polar phase, you should use high percentage of organic phases. So another thing you should remember that acetonitrile has a UV cutoff since most of the time we use UV detector for analysis. So UV cutoff is 190, but for methanol is 205. If I am getting a drug whose lambda max is suppose less than 205, and into my gradient illusion, if I am using a methanol, what will happen? As quantity of methanol increases, the absorption also increases. So in that case, you will never have a stable gradient baseline. Understand? So in that case, I will advise you to use gradient grade solvents, but again it will add a cost to your research. My aim for today's lecture is in low cost, you should do a novelty. Okay? So you have to give preference towards selection of molecule. It's lambda max. You should not waste the time in getting the drug and then you realize that it has lambda max less than 205. So most of the time, I am also getting a novel molecules have very less UV absorbance and they cause a lot of problem in HPLC analysis. Now here, a next slide, I tell you that most of the theses or paper, whenever they came, what they write is the molecular weight is determined using mass spectrometry and mass spectrometer gives molecular weight. So you understand that molecular weight and molecular mass are two different things. You should not use synonymously these terms. Now I will give an example. Suppose I will take my example. Okay, I am on Earth. Okay, I will measure my weight. So I will stand upon a weighing balance. I will note down 60 kg suppose or 70 kg. A hypothetical example. It is my weight. Now I will go to moon. What will be my weight? My weight will be zero because there doesn't exist any gravity. But my mass will remain same on Earth as well as on moon. So that's why mass spectrometry will give you molecular mass. But no doubt from that molecular mass you can take out information related to molecular weight. So if you see that, now here you pay attention. This is atomic number, this is a symbol, and what is atomic mass of carbon? 12.01. It is in 2 decibels. And how molecular weight of the substance is being calculated in next slide, I'll show you, and then you will understand how you can calculate in periodic table a molecular weight of the compound. So the compound of the atoms which have isotope, okay, you need to multiply their weight by their abundance. So carbon has isotope C13. Carbon 12 abundance is 99%. Means if you take 100 carbon atoms, 99 will have mass 12 and only one carbon among that 100 will have mass 13. So atomic weight calculated as follows. So here we have taken 12 multiplied by 0.99. How it comes? If you divide this by 100, it comes 0.99. So its atomic weight will become 11.88 and C13. If you do this and then you add this, okay, you get molecular weight. So you remember the compound which exists as an isotope, you need to multiply it by their abundance and then you get a molecular weight. But molecular mass, okay, and mass spectroscopy will be able to identify a molecule having a carbon 12 atom and a isotopic peak for C13 separately. Okay, and that's why this is called as mass spectrometry. No doubt, the atoms which do not have isotope, in that case, their molecular mass and their molecular weight are same. But if your atom consists of isotope, in that case, your molecular mass differs from molecular weight. And another important thing is, people write or students write mass spectrometry. Oh, sorry, spectroscopy. They write. So you understand, mass is not spectroscopy. Your UV is a spectroscopy. Your IR is a spectroscopy. Fluorescence is a spectroscopy. Mass is not spectroscopy. Why? For analytical technique to be a spectroscopy, you require a electromagnetic radiation to interact with your analyte. Okay, so in UV you have UV rays which are a part of electromagnetic radiation, so you call it as a UV spectroscopy. You have IR rays which interact with the matter, so IR is a spectroscopy. NMR, you have radio waves interact with the nucleus, so NMR is a spectroscopy, but mass is spectrometry. Okay, many a times in thesis we found a what? Mistake that student write that compound being analyzed by mass spectroscopy. No, you should write it is being analyzed by mass spectrometry. And see, what I feel is I also work into the industry. See, basics industry is in the need of the product. They are least bothered about the knowledge, chemistry involved. They want to comply with regulatory requirements. I do not criticize industry people. They are definitely absolutely doing a their job. Lot of them file ANDAs. Lot of them answers FDA raised queries related to impurity. But these basic things, okay, you only learn into the academics and you can apply this on a larger scale when you go to industry. Now here, okay, I'll tell you about isotope abundance. I'll tell you how mass is a very good technique in, okay, giving you molecular mass of the compound. So here, if you see this, one paper we published on lamotrigine and I got this, okay, three picks for lamotrigine. Here one, if you see, which is a tall. Here this is a second peak, and if you see this is a third small peak. Okay. So here if you see this, okay, these three peaks are there. So what is the exact mass of my compound? Exact mass is 256.0151. Okay, so it is blurred, but it is 261. I will tell you this is 256. It is 256.0154. See the accuracy of mass spectrometry. When you theoretically calculate exact mass of your molecule, it is 256.0151, and when you practically take out a mass spectrometer, so it is 256.0154. So first two digits or first three digits are exactly same, and there is a variation in last digit. So that is the accuracy, and nowadays a lot of journals are required to put a HRMS spectra. Why HRMS spectra? Because HRMS spectra will give you accuracy up to third decimal, and which will be very important when you characterize and establish a new structure. Now, why these are the three different peaks? Okay, this is a molecular ion peak. I am getting exactly same. Okay, this is my M plus 2 peak. So molecular weight plus 2, and this is my M plus 4 peak. Now, why this peak is being observed? See, whenever you get an isotopic molecule, you have to write this into the description. You know your chlorine has a two isotopes. Okay, one is Cl35 you have, okay, and another is Cl37. Okay, so when you have two isotopes, so in my molecule, I will get either about 35, 35. This is also 35, and this is also 35. So when I have both the chlorine atoms 35, I'll tell you that this is a 35, one chlorine, and another chlorine is also 35. So I am getting a molecular ion. But you know the percentage abundance of 35 is 75% and 37 is 25%. Means when you take 100 chlorine molecules, 75 will be 35 and 25 will be 37. So I may get m plus 2 peak. Why? Because one of my chlorine will remain 35 and my another chlorine may remain 37. See, my mass of the molecule increased by 2. Why? Because one is 35 and another is 37. 37 is plus 2 than 35. So I am getting another peak at n plus 2. Or there may be a condition that both of my molecules of chlorine are 37. Cl and 37. Cl. Okay, so in that case, see my mass, molecular mass earlier 35, 35, but now both of them increased. Okay, plus 2 and plus 2. So I am getting a molecular ion third peak at m plus 4. So that is the beauty of this mass spectrometer. When you have isotopes, you will get a isotopic peak. Understand? And that's why mass spectrometry determines exact mass of the molecule. And you have to write this into your thesis, into your paper. Most of the paper related to what mass fragmentation and analysis lacks only discussion related to chemistry. I will encourage you, just like somebody has encouraged me, to put lot of chemistry into your what work, and you will also enjoy this. Now you'll ask me, sir, why first peak is the tallest, followed by my second peak, and followed by my third peak is very short. Why? Because both 35, 35 is having 75% abundance. So this is a tallest peak. In second, 35 will have 75 and 37 will have 25. So its abundance is reduced. And third, both 37 will have 25, 25% abundance. So it is having less height. So this information, who gives a mass spectrometer gives. And so we should be very much blessed. I will say that it is my dream instrument that I should have a mass spectrometer. Okay, you must be knowing that mass spectrometer will tell age of certain substances depending upon their chemical compositions. So if you get some particular chemical species and if you find the composition using mass spectrometer, you can predict the age of that compound or that species. So in that way, okay, and slowly at present the mass spectrometric cost is only used, it goes to 1.2 CR or crore. Okay, but slowly as production increases, it will cost into the price of HPLC, then everybody will have what? Mass spectra on their table. So I hope that I made it clear that what information, okay, my mass spectra gives and how I should interpret to make the things clear that there is a difference between a molecular weight and a molecular mass. So we'll be going now. Most of the students ask me, sir, why we want an accuracy up to third decimal? Why? Okay, they are having a mass spectra, they are giving me 256. And is this matching with 256? Is it fine? Because I'm getting exact mass, but this is not the case. Next slide, you will come to know that even though species has, okay, first three digits are same, they are different species. So you cannot use only first three digits before decimal to establish your accurate structure. And for that purpose, we will understand the slide. If you see this slide will tell you this is a carbon. Okay. tell you. This is a carbon, this is hydrogen, nitrogen, and oxygen. And here is the list of the compound with varied composition. This compound consists one carbon, no hydrogen, one nitrogen, one oxygen. So there are varied composition, but if you see their first two digits, okay, if you see their first two digits, all are 58. Okay, all are 58. So if you don't have HRMS, see, but their composition, okay, this is having 4 hydrogen, but this is having 2 hydrogen. Their composition is different, but see their molecular mass into first two digit, okay, is the same. So my answer is you cannot use, okay, the mass spectrometer which is giving you. I get a lot of cases where they place only first three digit and they say that this is a structure of that compound, but no. You may have a same molecular mass, but your composition of the atom is different, and that is the reason you should have a third decimal accuracy into your mass. So if you see that when your compound is C3H6O, then and then only you will have 58.01. Their embedded value mentioned is, see the accuracy of mass spectrometer and show you in some of our research how we have been accurate up to fourth decimal many time to express our mass fragmentation. So that is the reason you require HRMS, and your exact mass should match at least up to third or fourth decimal. Then and then you will get an accurate composition of your structure and composition of your what elements or atoms into that structure. So this 58, but the structure is different. Okay, this 58, the structure is different. And that's why you have to go up to third decimal accuracy using mass spectrometry. Then and then you can predict most accurate structures. So I hope that I have given answer to many of your questions, whether we should go to what decimal, why mass spectrometry is a spectrometry, not a spectroscopy. Then we will switch to other. Now this is my drug candidate, and this drug candidate taught me lot of things. Okay, during a method development, many times it was a stumbling block for me, but okay, this drug candidate has taught. Okay, why I selected this drug candidate and tell you that this is a olopatadine structure. Okay, this is a formulation, Pantol. Okay, antihistaminic it is used. So new nucleus. And definitely when there is a new path, there is a lot of trouble to travel on that path. So new nucleus, no one has taken this oxypene nucleus earlier, so I took this. But definitely that drug molecule taught me a lot of things. No reported methods were available till we publish, because I told you that when you take similar methods, similar nucleus, you get lot of papers already available and you lose novelty. All reported methods were LCMS, but they are quantitative. No one has carried out degradation study on this. Most important thing is you need to develop a HPLC method. Then first step is selection of mobile phase and mobile phase pH for your drug molecule. Now it is always better to run official method. What students do is methanol water they run. If I ask them what is the basis to run, they cannot answer. Okay, they say that methanol is cheap, water is cheap. No, no doubt they are cheap, but you should have some rationale in starting somewhere. Either you should have some paper published where a lot of studies being done using that stationary phase and mobile phase, but arbitrarily you cannot take. And most of the time I found the method development part is only arbitrary. Okay. So it is always better to run official method. You tell that I have used British pharmacopoeia method, then a person who is judging you do not have any other alternative. Or maybe a IP method, because they are well established, they are well studied. No doubt, they will not exactly reproduce into your laboratory, but you need to what? Set up this method into your laboratory. First preference should go to methanol, acetonitrile, and buffer used, what I used is potassium dihydrogen phosphate. Okay. Now, these are the some basic things, but basic things, most of the HPLC things you know, I will not stress on PCMS part. Now here, why there is an adjustment of pH plus and minus 2, okay, plus and minus 2 of pKa value. Okay, everywhere it is given. Okay, why you need to adjust a mobile phase pH of plus and minus 2 pKa? For that purpose, you use Henderson's Hasselbalch equation. pH is equal to pKa plus log of ionized form to unionized form. And you know that pK is the thing where you get 50% ionized and 50% unionized form. And here, so you are not sure that where your ionized form will retain or unionized form will retain. But if you get a minus 2 pKa, then here you see that your unionized form is in 99%. And if you get plus 2 pKa, your ionized form is 99%. So either you go to minus 2 pKa or plus 2 pKa, you will have one form in dominance. Okay, so you require for HPLC separation one form into a dominance. Either you use ionized form or unionized form, but majority of things they do that since the stationary phase is non-polar, unionized form will retain more. This has to be put into the thesis that my drug pK is this, I have set up this pH is this, and you carry out study by using plus 2 pKa, minus 2 pKa and show that which form is having better retention in your stationary phase. But I learned this, okay, I understood this, but when I see my drug has 2 pKs, okay, again I faced a trouble, then which pH I should use? So whenever you are carrying out study, always do a literature. I did a lot of literature for this and I have received this patent. And in this patent, okay, this structure is being reported for this and they have given both the forms at what pH your molecule will exist. So before a pKa 4.1, my this cationic form was in predominant. Between pKa 4 to 9, I used to have zwitterion, both plus and minus. After pKa 9, I will have unionized form. So I have carried out study at different pH and I have selected pH 2.5, which is minus 2 of this pK value 2.5. Why? Because I found my cationic form gives me better retention and I have to put this into a discussion. Merely by taking arbitrarily pH will not solve your problem. So you have to put a lot of chemistry that what happens at that pH value, which form your drug exists and why it is retaining more. So I have put that cationic form has a better retention. Okay, cationic form has a better retention on my stationary phase, and I choose 2.5 as my pH. Okay, so now these are the routine things you will get. I will not waste on time how to carry out force degradation studies. Okay, so you have to take a drug sample, then you have to take a stressor, maybe acid or base. Okay, then allow to react, providing a heat for a sample, and then you neutralize this thing with what neutralizing agent. This now it is well established thing. Any literature will tell you how you can do acid hydrolysis, alkaline hydrolysis. Okay, they will tell you that. The only thing is important that you should not inject a substance into a column before neutralization because strongly acidic and strongly basic condition will leads to damage to the column. So if you see that, when I separated, I carry out stress degradation of my drug, I got This is the first base degradant. And then in photo degradation, I got second degradant, third degradant, fourth degradant, and fifth degradant. And then I characterize this all degradant by using LCMS. So now I wasted almost a 1 month to separate these degradants from each other. Earlier they were very close to each other. I was not getting a proper resolution, but my literature review helps me that I am using a cationic form and my degradants are close to each other. Hence, it came into my mind and through literature to use ion pairing reagent. Okay, there is a special importance of ion pairing reagent. When you use ion pairing reagent, they ion pair with the cationic form and they give a very good resolution to this ionic form. And when I use 1-hexene sulfonic acid as an ion pairing reagent, see this, my all degradants get well separated and their resolution is greater than 2. Now this again solved. Why the problem is not solved? Because I know the chemistry of molecule, what is happening at my pH of my mobile phase, and that's why I'm getting a solution. Once you understand the chemistry, you will get a solution automatically. So development of LCMS compatible method, I have used ammonium format buffer because it is volatile, which I told you earlier. You cannot use potassium dihydrogen phosphate or sodium dihydrogen phosphate. One important thing you should keep in mind, the LCMS apparatus which you will be using should also have a UV detector so that you can see your UV peak and the same peak you can send it for LCMS for further characterization. So I will advise some of the students take their samples, spend money, 2,000, 3,000 for injection, but they directly feed into their MS. They will not come to know whether their compound are being separated to that MS stream or not. So I will advise you that you should ask first operator that your MS should have a UV detector and you should observe a UV chromatogram and the same peak you should observe into that mass ion chromatogram. So you will come to know that which degradant is being fed to mass spectrometry and how you are going to characterize that degradant. Okay, so now first thing is you should calibrate mass spectrometer. If you don't calibrate, you will not get a third decimal accuracy. So tell the operator to calibrate. Then put a standard drug into mass spectra and see that whether exact mass tallies to the third decimal accuracy or not. This will tell you that whether your mass spectrometer is calibrated or not. If your pure drug, your mass spectra is not giving you accuracy up to third decimal, see to it that you will not never get this accuracy for your further drug fragmentation. So calibration of mass spectrometry is important. Check this calibration using standard drug and calculate error in MMU. So for accurate mass determination, 10 MMU error is acceptable. If your error is beyond 10 MMU, then that mass data will not give you accurate guarantee of the composition of your mass. Preferably use electrospray positive mode, or if there is a facility, run in both modes. Okay, there are mass spectrometer, you can run into positive mode as well as electronegative mode. So you can do this because you do not have to spend for another injection for same sample in electronegative mode. Okay, so these are the parameters you should keep in mind when you need to. So if you see that same drug olopatadine, my mass is, I have received this mass spectra. This is my molecular ion peak, and I'll show you a next slide, okay, how the accuracy is. So if you see this, okay, what is a practical mass? We can see this 338.1789. Now this is my theoretical mass. This is practical. Why practical? Because I have put this drug into mass spectra now. And this is a theoretical. I calculated using a chemical composition of C21H24NO3. If you see this, till first two decimal, I am getting accuracy 0.17 here and 338.17. And then I calculated error in MMU. What is the formula to calculate error in MMU? It is a difference between experimental mass and theoretical mass. Experimental mass is this, which you calculate via your experiment. Theoretical, which you calculate using pencil and pen. And if you see this is the formula, and I am getting error less than 10 MMU, okay, it is 0.38 MMU. So my accuracy of predicting this structure is very, very high. So that's why I advise you to calibrate. I calibrated my spectrometer. I calculated my error in MMU is very less. And then I carried out MSMS studies to understand fragmentation pattern of this molecule. So this I got a MSMS. These are all my mass fragments. When I put that molecule into mass 247, 231, then I need to predict the fragmentation pathway. Why? Because I will come to know how my molecule is getting fragmented into a mass spectrum. So here, if you do a study, this is my olopatadine molecule, and I am getting this fragment 203. So this is a 203 fragment. 191, I am getting 191. 178, 165, 178, 165 I am getting. So if you observe experimental mass and theoretical mass 203, 008, 203, 13, but error in MMU is less than 10. So whatever fragment I am getting for all of these fragments, the error is less than 10 MMU, and then I can predict the structure of this molecule, how it gets fragmented into mass spectrum. And I got a lot of trouble in predicting this fragmentation because no earlier reports were available for oxytocin ring system. But when I studied a lot of chemistry paper related to this oxytocin ring system, then in one paper they have given that this oxytocin 7-membered ring will get broken into this 5-membered ring, and then immediately I put a mechanism for collapsing of this ring system to 5-membered ring, and I got all the fragments which I observed into mass spectra with a accuracy of the error less than 10 MMU, and I can be able to predict the fragmentation pathway of the drug. Then what I did is I have put my degradation product 2 for again fragmentation. Why? Because I want to understand the structure of my degradation product 2. But unfortunately, what happened? I got a same molecular mass for my degradation product 2, same like drug. My drug is having same, my degradation product is having same. And also, if you look upon a fragment of drug and fragment of this degradation product 2, they are exactly same. So this was again a problem for me as, see, they are coming at two different places on HPLC, means definitely they are two different degradants. But their molecular mass is exactly same as a drug and their fragment is also exactly same like a drug. So we almost spent 2 days in repeating this mass studies, but HPLC shows a retention time different, but their molecular mass and fragments are exactly same like a drug. And then we will come to know that, okay, our drug was a Z isomer, and whatever degradant product 2 I am getting is my E isomer. So we predicted this structure of my degradant 2. So I come to know that in photodegradation, we got further literature that photodegradation gives you inversion of isomer from E to Z, and then we came up with this photodegradant 2 structure. Then, if you see that, then we characterize photodegraded 3 structure. See, therefore, here you see that olopatadine I got 338, but photodegraded I got 308 as a mass. Definitely 308 is less than 338, but my mass fragmentation for drug 203, 191, 171, 161 were exactly resembling with this degradant. So I came to know that this degradant is having some part less than the drug, but it gets fragmented exactly same like my drug. So by knowing this and by taking the help of drug degradation pathway, we established a structure for degradation product 3. Okay, this is our degradation product 3, where this ring gets collapsed and make a 5, and rest of the degradation pathway will be same like drug degradation pathway, and we came up with a novel structure of photodegradation. Now, here, the last two degradants are yet to be characterized, photodegradation 4 and photodegradation 5. So what happened is, photodegradation 5 mass is greater than the drug, and same mass we got for photodegradation 5. So we calculated that 338 minus 352, or reverse you can get, we are getting 14 mass unit greater to its degradant. So there must be a some group which must be attached to this, which is having 14 mass, but if you see their degradation products are exactly same for the drug. So this data will tell you that these molecules are same like a drug, but only some group has got attached, which is having 14 mass unit extra. Then we established a drug structure that carboxylic group, which we were having for olopatadine, got esterified. Okay, this is a COH, but it gets converted into ester. Why? Because in preparation of solution, we have used methanol. So methanol is alcohol, and this group is acid. Okay, and when we react in presence of acid, they get converted into ester, and one is a Z isomer. Okay, same like a drug. This is a Z isomer, and one becomes E isomer, and both of them got esterified. And that is the reason, okay, we have written into our paper that for the study of this drug, do not use methanol, but use acetonitrate, because if you carry out stress degradation studies using methanol, you are bound to get stress. So this information we reported, and I think that this was the first paper we published on oxygen ring nucleus, and we predicted degradant product 2 structure, which is its E isomer, then degradant product 3 structure, which get due to collapse of this ring system, degradant and photodegradant 4 and 5th, which happens, this got happened because of esterification of drug, and this got produced because of esterification of photodegradant 2. And I'll tell you that we will go back to our previous slides, and I'll tell you about a chromatogram. Okay, if you can see, this is our ester. Okay, this is, I mentioned that this is a ester. If you see, it is formed in less quantity, and this is the ester, it forms in higher quantity. Why? Because here this is a drug, this drug got esterified because of methanol. And here, this degradation product 2 got esterified because of methanol. So its concentration is always less because this isomer was available in less quantity. Its concentration is high because drug is available in high quantity. So this discussion we put into our paper, and we published it into Journal of Liquid Chromatography and Related Technologies. And I'll tell you that we received many appreciations for this letter, but this is not to boost our research. This is a simple research. What I will tell you is, you please select a novel nucleus and come out something new. Okay, that is my intention. That is not my intention, sir, that we are boosting our research, but that is to encourage you people. This is one of the scientists, okay, who was working in Teva Pharmaceutical Technical Library, okay, Hungary. He appreciated my paper, and at that time he has given me a call. Maybe he must be working on the same paper, and want to characterize a photodegradation product of the drug. So he talked me about 2 minutes, and we appreciated that. Okay, your paper is good. Please send me a reprint, and then you feel a good that somebody is recognizing your work. There is another mail I received from another scientist. Okay, that this is also a Blanche scientist. So what she is telling that I am very much interested in your paper. This is titled because I think there were very few papers or no papers which were done as stress studies and degradation characterization using this nucleus. So here I would like to acknowledge Dr. Sandeep Kale, who has helped me a lot in carrying out this LCMS studies. He literally used to stay with me at 2 a.m., 3 a.m., sometimes in the morning to characterize all degradation product. Heartfelt thanks to him. I think I'm crossing a time, so I will not take up a case study too. I will get this case study too if there was an extra time. Okay, but here on the last two slides, I'll tell you. Okay, these are also same related to mass fragmentation, but last two slides I will. So you need to predict mechanism also. Okay, whenever you put why the drug is getting degraded. Okay, so here also this paper we published into Journal of Liquid Chromatography. The drug is erlotinib used for lung cancer, and you need to say that in degradation this thing is getting okay degraded. Why it is getting degraded? So if you study a silver steel or a pavana, so you get a cleavage of the group which can produce a resonance stabilized structure. So here we produce that the cleavage happens to this group, and we are why the cleavage happens because the leaving groups get resonance stabilized. So this is actually acid degradation mechanism. So you try to put mechanism also and lot of chemistry also. This is also why this ether group is getting cleaved because of acid. So we have shown here mechanism. And we have shown also how base hydrolysis is being happen. Again, because of this resonance stabilization, how base is acting to what? To make another degradant. See, when you get a lot of queries from USFDA asks these queries, okay, what which geometrical isomer may be formed in degradation, what light will do, whether it will convert these isomers into one another, whether you can determine toxicity of these isomers, because you know when there is a change in isomer, thalidomide is a very good example. It suddenly any drug becomes a teratogenic. Okay, so with this here, I thank you all of you for patiently listening my lecture. Okay, so here I would like to stop. Okay, if you have any questions, if time is there, we will take up. Otherwise, you can pick up my email IDs or WhatsApp. You can send me. I will look upon if I don't know. Will search literature. If I know, I will immediately reply to you. So, I would like to tell that my coordinator that my part of presentation is over. Now, I would like to hand over mic to you.
Thank you very much, sir. It was outstanding and very informative. And there are some questions.
Yeah.
There are a couple of questions. Let me pick one.
Okay.
So there is one question asked by Minakshi Pandey.
Okay.
She asked if any product is not mentioned in any pharmacopoeial monograph, then how we can determine the limit of any impurities.
Okay, see, there will be a thing. See, impurity and degradation product are two different concepts. Okay, impurity related substance limit is given into the pharmacopoeia, but pharmacopoeia do not give structures of the drug degradants. So you actually need to carry out degradation study, establish a structure, and there is a ICH guideline. They have given a limit for degradation product depending upon daily consumption of drug substance and drug product. And then you can quantify by keeping that drug for stability studies whether that degradant is really exceeding to the limit or below the limit. If it is below the limit, you don't have to worry. But if it is exceeding identification, you need to subject it to LCMS, LCNMR, isolation, characterization. or if it needs to qualify, then you need to carry out toxicity studies. So I hope that this answer will solve the query to the student who has asked this.
Thank you, sir.
Yes.
Now I would like to invite Dr. Abhishek Tiwari, Professor, Devsthali Vidyapeeth College of Pharmacy, for the vote of thanks.
Yeah, good afternoon, everyone. I am Dr. Abhishek Tiwari, and I am here to present a vote of thanks for today's lecture. That is the application of LC and LC-MS for characterization of degradation products. I would like to thank our guest speaker, Dr. Anand Mahajan, for enlightening us with their knowledge. Today's lecture was full of knowledge and interesting things. It gave deep insights into the topic and also revealed some interesting facts. The points where Dr. Anand Mahajan told us about the regulatory aspects, including ANDAs, that is impurities in drug substances and product, selection of drug candidate, which includes categories like calcium channel blockers, proton pump inhibitors, antiviral and anti-hyperlipidoids, selection of chromatogram systems, and one case study discussed OLP, including validation parameters. The session was very informative, and I am pretty sure that the precious knowledge that Dr. Mahajan gave us will definitely help us and also our students for in their future. Once again, I would thank to Dr. Anil Mahajan for taking out time from his busy schedule and enlightening us with the knowledge. Thank you so much, sir. Also, thanks to all the participants present over here for paying their attention for such a nice and informative session. Thank you so much.
Yeah. Thank you, Abhishek. Thank you, Disha Dutta madam. And once again, heartfelt thanks to Principal Sir Ashok Kumar for a cordial invitation.
Thank
Okay,
you,
if any
sir.
queries, you can reach me. I have given my email ID to Sir. Any queries, you can contact me anytime.
Okay, definitely, sir. Thank you. And now, on behalf of Devsthali Vidyapeeth College of Pharmacy, presenting you a token of love. You kindly accept it and oblige us. It's a certificate for you, sir.
Thank you, madam. Yeah, thank you so much.
Thank you, sir. So here we are at the end of our 4-day technical sessions delivered by eminent dignitaries. And tomorrow, that is on 22nd January, the session will start at 10 a.m. We are going to have poster presentations by the participants, followed by prize distribution. The program will be graced by Dr. Narendra S. Chaudhary, Vice Chancellor, Uttarakhand Technical University, Dehradun. The link for the same will be shared half an hour prior. Till then, goodbye and take care. Thank you.